I was just saying at breakfast, he was one of the most productive scientists we had, but then he decided to leave us. So he was really good and equally successful at Panacea, where he later on went. So thank you so much, Jagat. I'm very happy to be here and really amazed at what you're building here. You know, this is just amazing from what you were telling me. You have 400 students there. You have, I mean, just amazing. I cannot picture you from being outstanding scientist in a company to now doing all of this administration. So congratulations to you. Very well done. Thank you, Ashish, also, who has been my local host. So what I'm going to do today is I've chosen a very generic topic because I know there's a lot of young scientists here. So I thought I will work on something. क्या हो रहा है मोबाइल? ओके ओके वापस कर देना है बस. Yeah, okay. Is it better now? Okay. This is, anyway, thank you for the generous introduction also. Yeah, so my topic today is to tell you about the emerging issues we have in heart disease, obesity and fatty liver. I'm pretty sure all of you are familiar with these words, and you probably know somebody amongst your friends or family who has one of these diseases. If they don't, eventually they will get one of these diseases. So it's a very important topic we're going to talk about. So yeah, these all these three collectively are what are called cardiometabolic diseases, which is cardiovascular disease, obesity, and fatty liver. So collectively, these are called cardiometabolic diseases. This is something that's growing all over the world. I'll show you some numbers. But as the world, or as people are living longer and nations are getting richer, this is a direct impact that you have. When you have a more affluent society, what happens is this is what you get. You get heart disease, you get obesity, and you get fatty liver. So it's a growing menace, especially in the developed nations, but also in India. I mean, I'm pretty sure you know a lot of people who have some of these issues. So. Where do I point this? Okay, I got it. Yeah. So just to show you numbers, these are global numbers. These are not just Indian numbers. 25% of adult population has what is known as hypertriglyceridemia, which is high blood triglycerides. It's a lot like high cholesterol, but it's a slightly different fat, triglycerides. NAFLD, which is non-alcoholic fatty liver, also impacts about 25% of adults. And I'll tell you the consequence of not having treatment of NAFLD. Obesity has just exploded. Over 650 million adults globally are obese. These numbers are pretty high in India also, especially in the middle class. And the unfortunate thing is we are seeing a lot of all of these things in younger population. In my parents' age, you would have heart disease when they were in their 60s or 70s, or you would have obesity in later stages. But you're starting to now see these diseases in young Indians in their 30s and 40s, and I'll tell you why that's happening. I am from the industry, and why are we interested? Because there's a huge market opportunity to launch products here. Each of them are multi-billion-dollar products, and so it's not just us. The next speaker is also working on obesity, or his company is working on obesity. Several people are working on all of these. So hugely attractive market opportunities for both large pharma and small pharma. So I just want to explain to you what each of these mean. Heart disease, which is nothing but heart attack or cardiovascular disease, these are different names given. Essentially what happens there is your arteries that supply blood, right? I mean, you have arteries just like water pipes that supply blood to different organs, especially when they supply the arteries that supply blood to the heart when they are clogged, which means they are blocked. You know, in your home also, the water pipes sometimes get blocked, right? Similarly, what happens in humans in the arteries due to plaque buildup, which is a buildup of literally fat being built up in the artery, the blood supply stops, and this is what causes heart attacks and stroke. We'll talk more about that. So that's heart disease, also known as cardiovascular disease, or causing heart attack and stroke. Obesity is nothing but an excess accumulation of fat in the body. Human beings are not, traditionally human beings were trained to accumulate fat because we were hunter and gatherers those days. We didn't know when we would get the next meal. So once we got a meal, we would eat a lot and store it, and that's where the obesity started to come. It's no longer the case now. We don't hunt and gather, we don't need to store, but that's the lifestyle we have gotten. So obesity is nothing but accumulation of excess fat. NAFLD is something new. This is non-alcoholic. There are two kinds of fatty liver. One is alcoholic and one is non-alcoholic. Non-alcoholic where is directly related to obesity. Most people who are obese will likely have fatty liver. The consequence of having fatty liver is if you don't treat it, eventually it causes cirrhosis, the liver fails, and you have to have a liver transplant. So all these three diseases are actually pretty detrimental to human beings. Even though they seem like very three different diseases, in our view they are all tied together. On the left hand side you will see triglycerides. I talked about high triglycerides, right? On the left hand side you will see high triglyceride. Triglyceride is nothing but composed of fatty acid on a glycerol backbone. It's literally fat and it accumulates in the artery. At the bottom you will see it's accumulating, that yellow hump that you see, it's accumulating in the artery, and basically blood supply is stopped, right? So that's heart disease. The second is obesity. You will see on the left hand side, and in normal anatomy the yellow is the fat. There's hardly any fat. We all have some fat, but what you see on the right hand side is gross accumulation of fat. Again, this fat is nothing but triglyceride. Triglycerides in the vessel wall, triglycerides in the viscera causes obesity, and the same triglyceride in the liver can cause fatty liver. So all along people have thought about this as three different diseases, but our hypothesis, and we have a drug discovery program I'll talk about, is fundamentally they are all related to triglyceride metabolism. You have to help me and tell me where to point. There it is. All right. So I've already told you that the basic component of all those three diseases is triglycerides. So that's one commonality between those three diseases. The other commonality is the risk factors. Risk factors is something that predisposes you to get the disease, are also very similar for all three diseases. Diet high in saturated fat, ghee for example, I mean it's a classic example of saturated fat. So if you consume diet which is high in fat, cholesterol and sugar, that's a direct correlation with obesity, hypertriglyceridemia and fatty liver. All three diseases have very common risk factors. Diet, lack of physical activity. This is beginning to be a huge problem globally. If you do not exercise enough, I'll tell you how obesity works and why physical exercise is so important. But essentially, poor diet, basically a very senile lifestyle or not having any physical activity. And so these two are controllable risk factors. And you and I can control our diet, we can control physical inactivity. A third component is genetics, which is what my grandparents had, my parents have. And if they were predisposed to getting any of these, I likely inherited those genes. That's something I can do nothing about. So two controllable risk factors, diet and physical activity, one non-controllable, which is genetics. So how does heart disease, I will talk about each of the diseases separately now. So let's start with the heart disease. How does heart disease affect us? It affects us in many, many ways. For example, when the blood supply due to clogged arteries by fat that supplies arteries to the heart is stopped, the blood supply, you get heart attack and heart failure. This everybody knows. You have the chest pain and all of that. But you also know that if the vessel wall involved is supplying blood to the brain, and if that is blocked or there is some clot formation, you get a stroke. You get vision loss if it's artery supplying to the eye, kidney disease and others. So fundamentally, the problem is the lack of blood supply to different organs, and it presents in different disease formats like the heart attack, stroke, blood pressure, the kidney. If the kidney blood vessels are actually clogged, it ends up in blood pressure. So it affects us in many ways, and oftentimes it's fatal, as you know. So what are the current treatment for heart diseases? The number one, if you go to a doctor, they'll say, do you smoke? Do you not exercise? Do you eat high fat diet? If you say yes, 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 they'll ask you to stop. If that doesn't help in 3 months, they'll put you on a drug such as called cholesterol lowering drugs called statin, right? So these basically statins are drugs. There are 7 or 8 of them. They've been around for the last 50 years. I was involved in the discovery of Lipitor atorvastatin when I was at Pfizer. Brilliant drug. Actually, statins have been a big boon to human mankind because the cardiovascular disease came down because of that. But I'll tell you, statins are now starting to have some issues. The last ultimate diet, medication, if none of that works, as you know, you have to go through a coronary bypass or angioplasty or other such surgicals. So these are the current approaches. Not all of them are great, as I will show you. Yeah. So what are the limitations of heart disease now? Statins, even though they are very good, we are now finding out that they predispose you to type 2 diabetes. So statins are, although they are good, but now after 50 years, we are now finding that statins can actually cause type 2 diabetes or at least predispose you. The other thing is surgical interventions. Not everybody can go a bypass or an angioplasty. It depends on if you're diabetic, if you're too old, if you're too weak. You cannot have those surgical interventions. So there are certain limitations. So the world is now thinking of newer ways of treating heart disease. So one of which I will just briefly talk about is something called PCSK9 inhibitor. So this is something that's just been launched. Basically what it does is it prevents cholesterol from being formed in the body. So as a result of which your blood cholesterol goes down and your risk for heart disease also goes down. This is something very recent. It's only been launched in the last 3 years by Regeneration in the US and certain other companies. To treat ischemic heart disease, which is when the myocardial cells are died, people are now thinking of using stem cells. So they introduce stem cells, so you regenerate those patches of the heart that have been damaged. So newer emerging treatments are here. So we'll have to wait and watch as to how cardiovascular disease fares in the next few years. Oh, sorry. Is this better? How do I go back? And the front is the other one. Okay. Right. Now I have told you about heart disease, the risks, and what's actually happening. Let me now tell you about obesity. Obesity, the best way to think about obesity is like a bank. If you eat more calories than you burn, which is you deposit more money than you use, right, your bank balance gets big. It's the same way. If you eat more calories than you consume, than you burn off, you will have fat put on. So it's a very, very simple, fundamentally very simple mathematics the body has. If you eat fat, use it. If not, I will store it and you will become fat. It's as simple as that. There is no other mantra to that. So what are the health risks? We saw the risks of heart cardiovascular disease. Now what are the health risks of obese people? Obesity by itself does not kill you. What kills you is some of the effects you see as based on having put on too much weight. Number one is type 2 diabetes. Most obese people will go on to develop diabetes. They also have blood pressure. They also have prone to heart disease. So, obesity is actually, I would say, one of the very few silent killers that can present itself in many different ways: arthritis, asthma, cancer, brain stroke. So, by itself, obesity doesn't kill, but what it does is it triggers certain other diseases that eventually can kill you. So, how is obesity currently treated? Obesity currently, again, the same mantra: they'll put you on a diet, they'll put you on a very strict, maybe 1500 calories a day type of a diet, and they ask you to come back in 3 months. If you have not lost any weight for whatever reason, then they put you on medicines. There are two types of medicines now. One is something called appetite suppressants. So, these, it's called Ozempic. This works on the brain, and it signals to the brain that you have had enough food, as a result of which you will eat less and less. The other is trying to block fat. See, the fat gets absorbed in the intestine, right? So, one of the medicines will block the absorption of fat in the intestine, as a result of which all the fat will come out in your feces. So, these are current two approaches vis-a-vis medicines. There is also an approach for surgery. You have all heard of bariatric surgery, where you basically your fat is surgically removed. There are many different ways to do that. There's 2 or 3 different surgeries. So, that's the last option that you have. Despite all of this, the world is saying increasing obesity that in the world. So, it's not just in developed nations, as I said, even in developing nations, you are starting to see a lot of obesity. So, the limitations really, lifestyle changes generally, especially when it comes to food and exercise, is not sustainable. Most human beings cannot sustain for their life a healthier, so that's a given. I mean, you look around and you will see people will get on a binge diet for about 3 months, then they get off the diet, put on all the weight back. They go to the gym for about 3 months, heavily exercise, they probably have their knee being busted and they stop exercising. So, it's very difficult to sustain a lifestyle of low fat and high exercise. Medications also, as has been seen, are not effective. Therefore, there's a lot of, in the pharma industry, besides cancer, currently I would say most amount of efforts are being put in obesity treatment. Several companies are working. Here too, as I said, surgery is not an option for everybody. Okay. I want to tell you a little bit about non-alcoholic fatty liver disease. As I said, if you consume a lot of alcohol, that gets into the converted to triglyceride, basically fat, and it gets into your liver. All right. So, that's the alcoholic, and in India, unfortunately, because the younger generation seems to enjoy alcohol more, you are seeing a big rise in that. But there's another version which is called non-alcoholic fatty liver disease. This is purely caused by having a high fat diet. Most obese people who are probably on a very high fat diet and don't exercise end up getting fatty liver. Essentially, there's an accumulation of fat or triglycerides in the liver. On the left hand side, you will see a pinkish brown liver, which is healthy, great blood supply. On the right hand side, you will see it's yellowish and you will see fat droplets there. This is the beginning of non-alcoholic fatty liver. What is the consequence, right? So, essentially again, it's fat accumulation. Heart disease, it was in the artery. Obesity, it's in the viscera. Fatty liver, it's in the liver. Right. So, what happens after fatty liver? Fatty liver, 3 things. The top is the healthy liver, the middle is the non-alcoholic, the early stages. If you do an abdominal MRI, the physician will tell you, sir, you have grade 1 fatty liver, grade 2, grade 3, depending on how much fat you have. After grade 4, it's irreversible. Most Indian men above the age of 50 probably, very, very likely have at least grade 1 fatty liver. They just don't know it, but I'm pretty certain that's what's happening. If you don't control non-alcoholic fatty liver, the end stage is cirrhosis of the liver. You see at the bottom there, the liver cannot function. And basically, at that point, once you have cirrhosis of the liver, your only option is to get a liver transplant, right? So while it might seem that I have a little bit of fat in my liver, so I'm okay, that's not true. If it gets really worse, your end stage, and your only, really the only serious treatment for that is liver transplant. So how is non-alcoholic fatty liver disease managed now? Unlike triglyceridemia, cardiovascular disease, and unlike obesity where there are drugs there, NAFLD does not have any drug. So it's purely based on risk reduction. What they will tell you is you get on a diet, they'll control your lipids, they'll control your fat, they'll control diabetes, because oftentimes NAFLD develops into diabetes also. So there is no good medicine. It's really a lifestyle change that they allow, but there is significant interest in this in the industry to develop new drugs for NAFLD. Currently, no drugs. hypertriglyceridemia alone, or with fatty liver alone, or with obesity alone. So based on the data we have, if the vaccine is safe, which safety studies are going on now, we expect to be able to launch this as a product for 3 separate diseases. You don't need 3 separate medicines. As I said, we are actively progressing this product towards human trials. A lot of safety studies have to be done, and we are currently doing this. With a little bit of help and luck, when I say help, we need some funding. Once we get that funding, we will be able to complete the safety studies and push it towards man. So I'm very excited about this. This is an approach that's never, what is called first in class. In the business, it's called first in class. It's never been done before, and at least in the animal models of disease I'm showing, our hypothesis of saying if we control fat intake through one target, we are able to improve all 3 diseases. So hopefully Jagat will invite me again next year, and I'll be able to tell you whether the product has moved forward or not. But one last message, since a lot of young people are here, I think this is a great opportunity being a pharmacist. Do not learn every single day. I mean, the future is yours. Biotech and healthcare sector in this country has never been better. When I graduated in science, my option was to either go join a bank or do an MSc, or I don't know, write an IAS on that. But your options today are many, many more, right? So make sure you make full use of your opportunities here. If you want looking for internship, I also run an internship program for my parents' foundation. We take students for 6 months, we pay them a stipend, and we train them in the lab and all that. So if any of you are interested, please do and we, unfortunately, this is only dedicated to women though, because my mom was a scientist, so I have dedicated the whole program to women. We have gone through several, so very happy to take any of you. Happy to work with Jagat and Geetam to facilitate that, but please, and our labs are in the CDFT, which is in Hyderabad, beautiful state-of-the-art labs. So I don't know whether you guys have to do internship or not, but if you have to, be happy to help. And like I said, it's mainly for women. Not mainly, it's only for women that we do this. So thank you very much for listening to me, and if there are any questions, I'll be happy to take. If not, thanks to Jagat again and Ashish for the invitation. I did not know what to expect when I came here, but I'm really enjoying myself. So I told the driver I might be going back home at 12, but I think I'll stay till 3 o'clock today. So yeah. All right, thank you so much. Yeah.
Thank you very much for your nice talk and for showing this opportunity to students.
Yes.
We have time for question, please.
So do you know the mechanism of action of your...
Just a second, please.
Yes, we do. I don't want to disclose it, but if we have a CDA signed between NVIDIA, right?
Yeah.
And us, I'll be happy to disclose. We are not going to do small molecule for this. So there are opportunities, as I was telling you at breakfast, is if you, this is a highly targeted, highly specific program. Happy to disclose that to you, but yes, fully. Yeah.
Thanks.
We've just, we're in the process of filing a PCT now, so yeah.
Thank you, Geda, for coming here and encouraging our students.
Thank you.
And it was a very nice lecture, and the lecture is perfectly fit for our students. I think there was not much complicacy in your lecture, and they all have understood what you have told. And you have extended that internship particularly for the female candidates.
Yes.
I appreciate that one.
100%.
I have one question
Sure.
regarding that vaccine. When the vaccine should be given? What is the right time?
Yeah. So actually, it's a really good question. So there are 2 circumstances. One is you are prone to obesity, right? I mean, you have family history of a lot of obesity or heart disease, or maybe fatty liver. Then as a preventive, prophylactically, you can take it. We have done that experiment, and it works even that. The other is when you already have some of these risk factors, and you start it, you're already obese. your BMI is about 30, whatever that metrics is, and your doctor says nothing is working for you, then you can get. So we are thinking both sets of population, those who are at high risk because they have family history, the others who already have these diseases, both of them should be able to take it.
So my question was that like other vaccines, these are preventary, means at the childhood we take some vaccines so that in future you don't get, but this vaccine is not like that.
No.
It can be given even after
Yes.
the person has accumulated.
100%. In fact, we expect major percent of our target population to be people who already have the disease and everything else has failed for them.
Okay, no.
Then they will introduce some vaccine. So yeah, I think that's the first set of population. The clinical trials will be done on that type of population. Yes.
Thank you. Maybe there are some more people.
Very good morning, Dr. Saxena. It was very nice presentation.
Thank you.
Actually, I want to know if, as per your rule, you cannot disclose what is the X, a kind of a tissue factor or proteins that is involved in triglyceride biosynthesis. Did you perform any kind of study because you are lowering the triglyceride levels? So subsequent effect on vitamins absorptions will be happen, as well as some of the hormones like cortisols, which are naturally in human, aldosterons, their synthesis will be hampered. Is that any correlative studies have been performed in order to find out that?
Yeah. So the vitamin issue comes in when you block the absorption of fat, because a lot of the vitamins you're talking are fat soluble, and the only way to deliver. So we are not blocking fat absorption. Okay. Fat will be absorbed, so your vitamins will be delivered. The only thing we are doing is stopping the fat from accumulating in the liver or in the viscera. So we are not. So vitamin intake or vitamin availability for the body should not be impacted at all. The other thing you asked was hypertriglyceridemia. What is
No, it was the those hormones like cortisol, aldosterons, kind of.
Yeah. No, there are existing medicines that lower triglycerides by 50, 60, 70%. That has not been a problem. So we are not concerned that if you lower triglycerides, you will have some of those issues at all. Same with cholesterol also. Cholesterol, people thought it's so important for every single to function. If you lowered cholesterol, something's going to happen. It didn't happen. At the end of the day, a physician will look at what is the risk versus what is the benefit. Yeah, if you have had 2 heart attacks, forget being deprived of vitamins. That's not the issue. The issue is, will you have another one and die, right? So that's one. So it's always a delicate balance between risk and benefit.
Another thing that if you're talking about like, so your vaccine is something not from triglyceride biosynthetic purpose. It was that the triglyceride which is deposited on liver. So is that the factor related to the tissue deposition of those triglycerides?
I'll just say yes and leave it at that. Yeah.
Okay. So okay, thank you, sir.
Yeah, yeah. By the way, our target is clinically validated, as well as has been validated by others too. See, not working on a validated target is very high risk, and a small company cannot take that risk. So, yeah. Thank you.
Thank you, sir.
Okay, we have time for one student question. Okay, over there, please.
Good morning, sir. My question is, how long would it take to translate this vaccine from animal models to human clinical trials, and how long would the trials go on for this vaccine?
Yeah, good question. So the next step for us is to do rat and dog toxicity. That usually takes 1 month. It's a 28-day toxicity. And then you have to take the results of all of what I showed and that to FDA or CDSCO, which is the Indian FDA equivalent. And then they will say, yeah, you can start phase 1 trials. So we'll have to go to phase 1 trials. Phase 1 trials in this case will probably be no more than 1 to 2 months. If it is, phase 1 is basically looking for safety. You're not looking for efficacy. If it looks safe, then you have to move it to phase 2. And since these indications are actually not like Alzheimer's, which is 10 years away, right? These are pretty rapid. So we expect the phase 2 clinical trials to be maybe 1 year or so tops. And if it looks good there, then phase 3. So all in all, if we are very lucky from today to get on the market, probably 5 to 8 years. 8 years tops, 5 years the earliest. So, yeah.
Okay. Thank you, sir.
Yeah.
Please join me thanking our speaker
Sure.
again. Okay, and the next speaker is Dr. Shrikant Ramachandran, who is highly skilled drug hunter and medicinal chemist with over 18 years of experience in preclinical drug discovery, having worked in both big pharma and biotech. He has made significant contributions to early stage drug discovery, ranging from hit identification to lead optimization and candidate selection. Dr. Ramachandran specialized in designing target-based medicinal chemistry libraries, parallel solution phase synthesis, and optimizing SAR trends and physicochemical properties. A major career milestone for Dr. Ramachandran was his co-invention of the novel ALK5 inhibitor, which is currently in the phase 1 clinical trials. His expertise spans diverse therapeutic areas, including infectious diseases, oncology, immuno-oncology, cardiovascular, CNS, inflammation, and metabolic drug discovery. Today, Dr. Ramachandran will deliver an expert lecture on the design, synthesis, and optimization of B-summit derivatives as CSF1R inhibitors for cancer immunotherapy, offering valuable insight into the development of innovative cancer treatment.
Now we would request our esteemed chairperson, Dr. Yurika Novak, and research scientists to come forward and felicitate our speaker, Dr. Ramachandran. So, felicitation.
Can you hear me? Okay. And this is the pointer, this one, right? How to go back? This one. Okay. Okay. Thank you so much. It's a pleasure to be here in the Geetham. First time in Geetham, even though I stay in Hyderabad. So. Thanks for the coordinators for inviting me, and happy to be in this campus and connect with Dean and other distinguished invitees and speakers. And since I've been here, I just want to talk about drug discovery in general, and just one case study to substantiate how industry does this business of finding drugs. from my own experience. So just but the difficult part about industry is we can't talk many things unless it is published, right, in the public domain. So I'm going to talk about a published material. So happy to take you through that journey. So before I begin about the talk, I just want to give some brief. I think there were questions about drug discovery or new medicines. So this particular slide, it's one of my favorite slides. Whenever I go to talk in India, I'm going to pop it up because it's so relevant till now. So this is the analysis of new drug origin, from where the new medicines comes to the patients, right? This is analysis done sometime back in 2010, but I think it's still very relevant and it is not going to change this landscape. So if you look at this distribution of new medicines coming to the world, so 50% come from United States of America, right? So that's where the innovation happens. And the rest 50% is distributed across mainly the European countries like Germany, Switzerland, because there are some big pharmas out there, UK as well, and there are around 10% from Japan, right? And then the rest of the Europe. And you might be asking, oh, where is India here, right? So if you look at the rest of the world, which contributes the 2%, India, China all comes there. So the biggest population countries are not innovating here, right? And innovation comes with a cost, right? This innovation doesn't happen overnight. It comes with an extreme cost. And for these developing countries, probably you can't afford it to pump in that much money because there are a lot of failure. This is a high risk, high reward industry. Of course, it's a business, right? So, and one more interesting analysis from this same publication is this on the right-hand side. This is more striking for an academia. What it shows is that where these molecules came from, okay? Who discovered it first? There are two parts. There is something called drug discovery and a drug development, right? So if you look at this, in the United States, the large pharmaceutical companies contribute around 50% of these new inventions. But the striking features are this one. Around 60% came from small biotech or academia. That is, the original discovery happened in a small company or an academic professor, which was in-licensed by a big pharma for development. So this only happens in the United States. That's why that country is so innovative, right? So there are people, irrespective of where you discover it, they are ready to take those innovations forward, okay? And if you look at other countries, it's still the big companies do the heavy lifting, right? Whereas in United States, I mean, nowadays the big pharmas are just development companies or marketing companies. They don't do much of innovation because of the cut in the R&Ds and stuff like that. So biotech is the new world where the new drugs are coming out. So this is very striking, and I love this slide because of the the interesting features out here of how and ever drugs have been developed across the world. And next slide, I just want to, since there are a lot of students, I just want to show you how a drug discovery development process works. So this slide shows, so there is a phase called preclinical research, where the basic research of the mechanism of action of what target one should work on, the disease biology, that can happen in an academia setting, right? The basic research of doing gene knockout, knockdown studies contributing to disease can happen here, the basic research. And then the companies start looking for, making inhibitors or, down-regulating or up-regulating the protein or gene of interest, and they start picking up those concepts, right? And most of the drugs, the most convenient way of drugging people is by pills, that by oral route, right? So many of companies will start finding oral compounds out here in the drug discovery phase. So there is where, the chemists get involved, making compounds, doing structure-active relationship, getting it better, right? And then it goes into a preclinical battery of testing, which involves, safety pharmacology, both regulatory and non-regulatory testings. And then, as Dr. Uday mentioned, it goes for a regulatory submission, which is called IND filing out here. So if you look at the role of medicinal chemistry here, it's very small, right? it's in the upfront area where, you know, maybe in the first 2-3 years, you have a role of medicine and chemistry, right? Then you fix on one particular compound, even though we make hundreds of compounds or thousands of compounds to begin with, you ended up with one being selected here, and that goes into a process development, that goes into manufacturing, starting from milligram to gram to kilograms and tons to support all these following activities. So your phase 1 trial starts here, again looking at healthy volunteers, which can be done in a tens number of people out here. Phase 1 typically costs runs less than a year sort of phase, and then if where you look at basically safety and pharmacokinetics in humans, right? How does your compound behaved? Is it as predicted, using a dose to human prediction, or is it can you reach up to the can you do a single ascending dose safely or multiple ascending dose safely, and what is your limitation, and what is your expected efficacy dose, right? Are you reaching it based on your prediction? So all these are done here in the phase 1, and then you start picking up some patients out here in the phase 2. So here the volunteers can be hundreds, and typically it runs depends upon the disease area. It runs for a year or two, depends upon the number of patients you pick up, the diversity, ethnicity, etc., right? And then this is where all the drugs getting killed, right? Phase 2. Phase 2 is where you know whether there is a meat out here, right? And both the first in class and best in class get to know here in phase 2 trials. And that's, if successful, it moved into a bigger, larger population of phase 3, which is thousands of people out here, which is a registration trial. And then it goes into the NDA submission, and there is a FDA review happen, and the medicine is getting approved. So this entire process, right, it can run into in a long time, like at least 10 years from the very beginning out here, and that's a kind of an average minimum time, I would say. There are drugs which came after like 20 years from the basic research. So, but how much? The question is how much, right? I mean, I'm not talking about one compound here. This is considering all the attrition happened all the way. So you might have started like 10 programs or 20 programs out here, and you eventually ended with one. So it's all the attrition, considering all the attrition happened to that portfolio. This is the cost, right? Nobody invents a drug from the first program. That's unfortunately the truth out here. So it's a very expensive affair, considering all the attritions and failures happening in this industry, and it's very lengthy process. So that's why we being a culturally risk-averse society, we don't invest on this high risk, high reward industry. Okay, so now I'm going to talk a little bit about the preclinical research, which I was heavily involved in the preclinical research. So this also you can split up into different categories. So starting from the target discovery, and then that is the basic research of the target protein. You want to look at it, and you gain confidence by doing a lot of experience out here, whether it's a good target to use or not. And then you want to find a molecule which blocks that target mechanism, right? That is the hit discovery. This can be done in many different ways. If it's a big pharmaceutical company, they run high throughput screening against the target of interest using hundreds of millions of compounds out here to find the best hit out here. And if you are a small company, and if there is a prior knowledge on the target mechanism out here, you can get protein crystallography data from the public database like the PDBs, and then you can do molecular modeling on the compounds of your interest. Then you can do virtual screening, and you can still find a novel hit out here, which you can progress into lead identification. So the medicinal chemistry basically plays out here in the optimization process of the hit. Hit is basically an active compound on the target, but it's unoptimized for its drug properties. So you need to build in a lot of drug properties to make it orally bioavailable compound. So that's where the role of medicinal chemistry happens here in the lead, and then kind of lead optimization also. You do a lot of medicinal chemistry where you fine tune it to be the best if you are targeting a best in class drug out here. So then it goes into animal testing. One case study already shown here. You can test most of the models people pick in industry are mice model, which are one of the better translational models. So you can test it in the mice. You create a disease in the mice and then try to treat it with your compound. Single dose, multiple ascending doses just to see a dose response in the animals, and then you gain confidence and you can run through, just like Dr. Uday said, you can run it through multiple animals also if there is a models available out there, and then you do a lot of safety testing in the animals, and FDA required rodent and non-rodent species safety. That is basically you pick mice and rat as rodents and dog or cyno as species for non-rodent. So once this is done, it can move into some trials. So now I'm going to talk about a specific disease area, oncology. So oncology has seen a big shift in the drug discovery landscape all through these years. So starting in the early 1980s, when chemotherapy was the only available option for the cancer patients. So again, chemotherapy is not as safe, as you all know. They are not tumor specific. It kills all the rapidly dividing cells, and it's highly toxic. But it has been one of the mainstay in cancer for a long time, right? And then comes the 2000, where we started getting targeted therapies. So this is where the targeted discovery happened, where people start looking at overexpressing proteins or targets in cancer cells. Again, cancer is very heterogeneous. So for given a within cancer, you will try to see a lot of overexpressing proteins targets, which could all can become a drug discovery target mechanism. So that's why the targeted therapy looks care of specific mutations and overexpression in cancer, and then people started trying to make compounds to block it. So all the kinase discovery happened in this era of 2000, starting from the Gleevec, and right now there are around 80 kinase inhibitors approved in the clinic. So then in that 2010s, we have the immuno-oncology start to popping up. So what is immuno-oncology? Immuno-oncology is trying to kill the cancer using your own immune cells, right? But when cancer happens, what it really happens is that it suppresses many of your immune cells. So they trigger a lot of overexpressing receptors on those immune cells, like your T cells, myeloid cells, your natural killer cells, and those won't recognize cancer cells. So when cancer happens, it creates an environment not to identify them, right? Then scientists become smart to identify those receptors which are expressed on the immune cells and try to block them so that the immune cells can go and recognize the cancer cells. It's a very interesting area of research, and this immunotherapy is getting rewarded now. And now we have drugs out there market targeting this immunotherapy. And the good thing is that it acts like a vaccine, right? Once it starts recognizing, and your relapsing is it's difficult. So immunotherapy, but the problem is that it's so heterogeneous. The immunity is so heterogeneous among the patients. It doesn't work for everybody. It only works for a subset of patients. So there is a lot of work to be done in the immuno-oncology area because of this heterogeneity. So today I'm going to talk about a little bit about the IO program which we have run recently, and again combination of a lot of IO is need to be. So the goal of IO is to engage the immune cells, basically go and attack the cancer cells. So there won't be much toxicity per se, but you can get immune related adverse effect. Okay, if you are upregulating immune cells, and so there are kind of cytokine syndromes, storms can happen, but you can control because you are dosing pills, right? You can control the doses and you can get away with that. So okay, oops. Yeah, so this is some of the publications where this immunotherapy has been discovered. So there are two main stage immunotherapy receptors out there: CTLA4 and PD-L1, PD-1, PD-L1 checkpoint inhibitors. And now you also have the CAR T cells therapies out there. So again, the hallmark of immunotherapy is the durability of the response, and it acts like a memory for the adaptive immune systems, which translate for long-term survival for those who are benefited from this therapy. So again, a lot of these therapies have been approved now. Okay, it's a problem. So this is some of the approvals. You can see very lately in the recent years there are a lot of approvals in this cancer immunotherapy agent, but unfortunately these are all monoclonal antibodies. So there is no small molecule immunotherapy approved so far. So you need to get infused these drugs in a hospital setting. Okay, and in 2018, American immunologists got Nobel Prize for discovering some of these receptors, like James P. Allison and Tasuku Honjo received a Nobel Prize in physiology or medicine for inventing these receptors expressed in these immune cells. So this has been rewarded, this industry. And yeah, why we need additional immunotherapy? So people might ask, okay, you already found 2 receptors, right? Why we need more? Because I told about the heterogeneity out there, and if you look at this spreadsheet of different type of cancers out there, and if you look at the non-responders for the same PD-1 inhibitor, so only few patients are benefited from this therapy. Because of the immune heterogeneity, you have cold immune system, you have hot immune system, and these receptors are not expressed in some of these patients. So with one receptor, you are not going to treat the entire patient population. You need different type of immunotherapy which are targeting different receptors on those immune cells. So you basically need a combination of immunotherapy to treat a large set of population. So that's why I'm going to talk about another mechanism by which you can target some of this immunotherapy. So there is tumor associated macrophages or TAMs. They are also highly expressed in the cancer environment. There are 2 types of macrophages. There is M1 and M2 macrophages, and M2 macrophages which are pro-tumor macrophages. They express a lot of receptors, and one of the receptors we were interested is the CSF1R, which are expressed in these myeloid cells. And they expressed in these M2 macrophages, not on the M1 macrophages, right? You need to convert these M2 macrophages into M1 so that they can help the T cell expression, which will in turn go and target the cancer immune cells. So why we were interested in CSF1R is, it works through a kinase mechanism. It's a serine threonine kinase. So as you know, there are a lot of kinases being discovered as drugs. So medicinal chemists can easily target the ATP binding domain of kinases. So that's why reason we were interested in the CSF1R program. So CSF1R is a key regulator of the M2 macrophages, and again, as I mentioned, it's a type 3 protein tyrosine kinase family. So one can easily identify, and this is an old slide deck. So right now this number stands at 80 kinase inhibitors approved by FDA for different type of oncology indication, and some for inflammatory conditions as well. So kinase has been a mainstay for targeted therapy, both in oncology and in some inflammatory conditions. So coming back to the CSF1R, there are a lot of work happened in this area, and there were some of moved into clinical trials, and there was one compound been approved. In fact, this compound PLX3397 was approved for a small rare cancer where the CSF1R expression was higher. So it acted like a targeted therapy out there, and there were some other companies like Novartis had this compound in clinical trials, and J&J had this compound. So all targeting the ATP binding domain of the kinase. And also there are a lot of monoclonal antibodies. You can target the CSF1, which is the ligand which binds to CSF1R. So you can have opportunity for monoclonal antibodies as well, targeting the CSF1R blockage. So our lead generation strategy was very simple. We were looking at the literature, look at the starting point for this program, and then we found this interesting compound out there in the literature. This was a previously reported kinase inhibitor in the literature, but this was not very specific for CSF1R. It was hitting multiple kinases. Our aim was to make it more selective so that you don't have any toxicity issues coming from this scaffold. So we started this program looking at this compound, which was a known compound, and when we checked this in the biochemical assay, so we had 2 assays. This is a target assay, which is a CSF1R kinase assay, and this was an off-target pharmacology for the same compound c-Kit, which was giving toxicity for this program. So, we want to get away from the c-Kit, we want activity only on the CSF1R biochemical assay. So, this compound was not very selective at that point, and then we started working on the SAR on this compound. So, you can see the first thing was we looked at this particular group here, start changing it into different groups. So, you can see we started with a thiophene out here, and then we kind of looked at these other groups like this R2 and R3 on the periphery, and when we changed this R2 group from methyl was and especially R3 was the most striking. The R3 was initially started like CF3. When we changed the CF3 to hydrogen, we lost all the activity out here. So, then we realized that this is very critical for this compound. So, we kind of kept it constant, and then we also start changing the other groups. So, like here the chlorine is good instead of the methyl along with the CF3, it retained the activity, and this is a mouse macrophage. This is a mouse macrophage. This CSF1R expressed in the macrophages, right. So, we have two cell lines. So, this is a human macrophage, bone derived macrophage, and this is a raw macrophage as well. So, we want activity on the cell as well, because this is an intracellular target mechanism. So, we want cell permeability also. So, look, going through the SAR, you can see one can get away from the thiophene, which is metabolically labile group, and to something like metabolically stable pyridine groups out here, which retained the activity. The good news is that it retained the activity, but we did not see much of selectivity against c-Kit at this point, and there was some cell permeability as well, this compound, whereas some of this compound did not work on the cells. So, we moved on from this scaffold further. We did some more SAR here, basically looking at these amide groups. So, there are two amides in this compound. We started looking at the essentiality of this amide. We tried to modify this amide, but it was kind of losing activity on this, and then we tried to look at this amide group. Again, it was kind of losing activity. So, we kept this amide constant, but the one good thing is that you can reverse this amide here. So, you can have this kind of configuration out here. It retained the activity without losing the potency, right. And then we looked at the amide ketones, the essential amide ketones. We taken away one amide ketone, which start losing activity. We taken out the second ketoamide, that's again losing activity. So, both the amide ketones were essential for this compound to be active, along with this CF3 group. So, we learned a lot from this, and then we zoomed into this kind of a scaffold now, where the amides are fixed, the CF3s are fixed, right. And then we did some more work on this R1 group, which was very flexible. So, we could start seeing a lot of highly potent compounds here when we started working on this R1. So, we can get into picomolar potency out here on the CSF1R, and then we started giving good selectivity, because we improved the potency on the CSF1R. So, we started moving away from the c-Kit with something like great selectivity out here. Again, that is basically working on this R1 site, and we started seeing cell permeability on the BMM cells. So, that is the good news. So, we are having potency, we having selectivity, and we are having cell permeability. So, great place to be in. And then typically as a pharma company, you want to know the pharmacokinetics, right. We do not get into animals directly, we do some in vitro assays to looking at the metabolic stability. So, for that we selected these two compound type 13 and 14 because they were very potent and having cell permeability. So, we started looking at the metabolism and oral exposure. So, here typically a pharma company look at the in vitro metabolism first, looking at human and mice metabolism, which mainly driven by cytochromes, right. So, we did some in vitro ADME work. We looked at the kinetic solubility. This was some high log P compound, so the solubility was not very good, it's only 4.7 micromolar, but when you looked at the human and mouse microsomes, they were poorly stable. In human it was like 50% stable after 1 hour, and in mouse everything was gone when we checked, it's only 7% remaining. And the plasma was not a big issue, both in human and mouse plasma. So, it was mainly a cytochrome mediated metabolism, and we want to find out where it is happening. So, we did a meta-ID, to find out where this metabolism is primarily happening. So basically when we look at the meta-ID incubating with this compound with mouse microsomes, we found there is a demethylation happening here on the piperidine ring, on the piperazine ring, and then we get this compound, right? And interestingly, we tested this compound also. This is a metabolite. We also made this metabolite. It was active on the CSF1R and have some good selectivity, but it was not very cell permeable compound. And you can see the microsome stability improved, right? So you can see from here it was poorly stable, but this is a much stable compound out here because it's not furtherly metabolizing here. And in order to make sure that there is a good in vitro to in vivo correlation, we dose this compound into a PK study, right? You always want an in vitro to in vivo correlation. The models you are working on in vitro, you want to make sure that it is validated. So we did dose this compound both on IV and a PO arm. IV arm basically tells you the clearance mechanism, how fast and what's half-life of your compound. PO basically tells your oral bioavailability, right? So when we dosed IV, we see a high clearance, and that kind of correlates back onto this compound. So we dosed this compound number 13, and on the PO, the oral absorption was very poor. We got only 11% in a mice PK study. So we know that this is not optimized. We still need some work to do on this compound. So again, we went back into modeling. So there are some crystal structures available for this compound in the public database. So we want to look at the the modeling thoroughly. So when we model this compound number 13 out here, so there are two types of because this binds to the ATP domain, right? But even in a kinase inhibitory class, there are two types of kinase. One is the ATP competitive kinase inhibitors you can have, which is a type 1, and you can also have an ATP non-competitive kinase inhibitor, which is like a type 2. And you can, and a third one, you can have an allosteric binder, which is not binding to the ATP binding site, but still blocking the ATP mechanism, right? So there are two crystal structures available both for type 1 and type 2. Our compound modeled very well on a type 2 type crystal structure. So we used this DFG out conformation modeling, and then when you look at the modeling, we did some interesting observation here. We kind of not seeing the typical hinge binding. The hinge cysteine was the hinge binding residue was the cysteine 666, and unfortunately in this compound we are not observing that engagement of cysteine binding, the hinge cysteine getting engaged in this model. So we were like very, very amused with that because it was very potent compound, but we had some good additional interactions on this compound, and this was a solvent exposed area where this solubilizing group was moving in. So then we want to thinking about engaging the cysteine 666, we designed couple of compounds to do that. So we know from our previous meta-ID that this is a problematic group where we are seeing a lot of metabolism. So we were thinking, can we remove this group, and and then definitely we might lose some potency because there are interactions out here, and can you balance that interaction by bringing the cysteine interaction into play. So we did some modeling there. Okay, what happened? Yeah, so we designed these two compounds. We have taken out that the piperazine group here, right, in both the compounds, and we brought in a cysteine engaging group, this pyridine nitrogen, in these two compounds. Okay, and then the binding scores were very good on this compound, even though we were missing this big group out here. But this is just modeling, right? You need data to prove that this compound is a good compound. So we made this compound. So next is synthesis of this compound and testing this compound. Okay, we still have problem. Is it here? Can someone move the slide? Oh, I have to show here. Okay. So yeah, so this is the compound initially we docked, and we kind of showed this data earlier, right? The compound number 13, and so this has an issue with the metabolism, right? And then we kind of we made a one point change in between. We made this compound. We have taken out this compound, this group out here. You can see how much drop of potency you see by removing this group out here, right? So, this was important, and we dropped the potency, right. And then we knew that we were not engaging the cysteine out here. So, that is the reason we brought back the pyridine in here. So, you can see just by introducing one nitrogen out here, we gained so much of potency. We moved from 300 nanomolar to picomolar potency just by doing one change, one atom, and you still do not have this group here, right. The modeling also showed that this could have been a good compound. So, it really turned out to be a good compound just by doing this change, right. And just to substantiate that, we made one more compound by putting one more nitrogen here, which was not picking up any interaction. So, it kind of retained kind of that potency, and the good part is that by introducing nitrogen into the aromatic ring, you reduce the log P, you increase the solubility of the compound. So, we introduced one pyridyl in here which picked up the interaction and gained the potency. We introduced one more nitrogen into the ring, and where we gain back a lot of solubility on the compound. And the good news about these compounds are they having good selectivity over c-KIT. It has good cell permeability, that is very important, and it start giving some good metabolic stability to the compound. So, we started tick boxing many of the desired features on the compound. So, that is how we ended up with these two compounds. So, by removing this group, we achieved these two compounds, compound number 22 and 23, our best compound so far in this project. And then again, these are in vitro numbers, you need data on animal, right, that we kind of tick marked most of this. So, then we did some more ADME studies on these best compounds, compound number 22 and 23 out here. So, compound 22 and 23 taken out for the CACO permeability. CACO permeability is an in vitro assay where you measure the permeability, the potential gut permeability of the compound, so that when orally dosed it can absorb into blood. So, when we looked at these two compounds for the CACO2 permeability, so you can see compound number 22 had issue. It has it was a good permeable, no, compound number 22 was good. It was having high permeable on the A to B front, and B to A was lower, so that the overall effects was around 2.6, whereas compound number 23, which was permeability was poor, and the efflux was big, right, it was like 81.5, and then that is the reason we decided to test compound number 22, which was having better metabolic stability and better permeability across the CACO2 permeability cell lines. And we tested this compound in a PK study, again similar IV and PO dosing, 2 mg per kg IV and 10 mg per kg PO. So, here you can see the clearance came down as expected, as predicted from the microsomal data, and in the PO dosing based on the permeability, we improved the oral permeability and we improved the oral absorption. So, it is around 64% orally bioavailable compound. We had a decent exposure AUC out here, and this compound become our lead compound after all the optimization. So, unfortunately this program did not move forward at this point. We kind of we wanted this to take this compound further into the PD model, because now we had a good compound in our hand, which is a novel compound with IP over here, but unfortunately at this point the program did not move forward due to strategic reasons, and so we did some more work just on the biology front, because I mentioned about two type of kinase inhibitors, right. One is a type 1, which is a ATP competitive inhibitor, and there is a type 2 which are ATP non-competitive inhibitors. So, to prove that, we did some mode of inhibition studies using the kinase assay. So, there was a reference compound out there which is a Novartis compound, which was an ATP competitive inhibitor. So, we titrated this compound with ATP. So, if it is an ATP competitive inhibitor, you will start losing your potency by titrating with ATP. So, you can see the BLZ compound, it was having potency out here on a normal ATP condition, but you increase the ATP concentration, you got a right shift on the IC50. That means the ATP was pushing out your compound out here. So, this was our positive control. These are the ATP competitive inhibitor type compounds, and when we looked at our compounds, these are two our compounds, when we increase the concentration of ATP, we were not seeing the right shift on the IC50. So we were like in ATP non-competitive inhibitor. So that's how we differentiated ourselves from the others. So we really ended up with an ATP non-competitive inhibitor, unlike the Novartis compound, which is having good potency, good oral exposure. Yeah, and that's where this story ends, I would say. Yeah, this is just a synthesis slide for people who are interested in synthesis of this compound. This compound were made by this kind of a general synthetic scheme. Yeah, this was just an acid-amine coupling out here, and then you can do a lot of Suzuki coupling out here using boronic acids, and yeah, similarly you can make a lot of library of compounds. Not a a tough synthesis, easy to make compounds, and so, in summary, we started from a very weak non-selective CSF1R inhibitor, which are having activity on c-KIT as well, and many other kinases, and low metabolically stable, high potential for PGP flex, and poor oral bioavailability. And then we took that compound forward, did some SAR, which I explained now. Now people know what is mean by structure-activity relationship, right? You kind of find out the best part of the molecule, you remove the unwanted part, keep the best, and came up with this compound number 22, which was very potent and improved metabolic stability, good permeability, and efflux, and improved oral bioavailability. So yeah, that's where this ends now, and just want to thank the team who made this discovery. This was a collaborative work between Medivation, which is now Pfizer, discovery team, and this was a company I was working at, Integral Biosciences, Noida, and both this team collaboratively worked on this project, and of course many other projects, but this is one of them. Yeah, we had some good success story which has also moved into phase 1 trials, but this is another program. Probably I can talk about that compound in a later date. So, and thank you so much for your attention.
Okay, we have some time for discussion, for your question.
Yeah, very nice work, Srikant. So I think you guys made the right call. That drug was not going to be an oral product. But did you think of pursuing, I saw that the IV looked pretty good. So was the mandate only to go after oral, or IV was not on the table?
Yeah, IV was not on the table because there were other competitive oral CSF1R inhibitors there. So
okay.
Yeah, we want to be competitive with an oral drug.
Yeah.
Yeah.
I think the only way that drug could have moved forward is if you wanted to dose 4 times a day or something.
Yeah, yeah.
Right, because the T-half and was so low, I think less than 2 hours or something.
Yeah, yeah.
Yeah, so you need ideally 8 to 12 hours is what you need for once a day.
Yeah.
So you would have had to dose. Yeah, nice work.
Yeah.
Yeah, congratulations.
Yeah. We just want to do some PD work with that compound. Yeah, but due to the strategic, because this company was acquired by Pfizer.
Right, right.
The parent biotech, so yeah, they kind of scrapped all the programs.
Medivation is originally Russian, right?
No, it's a Bay Area based company.
Okay.
Yeah.
Yeah, very nice.
Yeah,
thank you.
Thank you.
Yeah, very nice presentation. Since I was also involved in SAR studies, just a few things came to my mind that that piperazine demethylation probably you could have avoided with morpholin. Was it?
Yeah, I think we made morpholin also, but at that point our aim was to improve the kinase selectivity and the potency, right? The hinge binding area. Yeah, I think we made the morpholin analog also. Yeah, and the molecular weight was getting bigger, above 500. So that was like another concern, how to reduce it further and keep the minimally required pieces, right? And the log P was another concern, like the solubility, right? So that's why we kind of make it short. We kind of removed it. Still, we achieved a lot of potency, right? And good solubility and good permeability. So it's also kind of a balancing act.
Yeah, thank you for a nice presentation.
Any questions from students? because I think there were queries about the drug discovery paradigm, so.
Really? Students, no questions? Okay, then let's thank our speaker.
Thank you.
Again.
Now this marks the end of session 5. We'll now take a short break for high tea. Please reconvene in 15 minutes. Please be back by 11 o'clock. introduce the chairperson of our next session, Dr. Uday Saxena. Dr. Saxena is a renowned expert with extensive experience in pharmaceutical and healthcare industries. His exceptional leadership and expertise have contributed significantly to advancing various medical and scientific fields. Sir, please grace the dais.
Okay, good afternoon, everybody. Can you hear me? So I have the honor of introducing the next speaker, Dr. Gangaraju Geda. Dr. Gangaraju is a recognized leader in the fields of nanotechnology, biomedical research, and sensor technologies. He currently serves as the International Relations and Research Advisor and Brain Pool Professor at Chwang-Ang University. Did I pronounce that correctly? I hope so.
Yeah.
South Korea, as well as an Associate Professor of NITTE University, India. Additionally, Dr. Geda is an editor for Wiley JNT and Frontiers in Drug Discovery. Along with his academic roles, Dr. Geda oversees medicinal chemistry initiatives at Envida Biosciences, where he drives cutting-edge R&D efforts to advance compounds towards clinical development. With over 20 years of experience in medicinal chemistry, Dr. Geda has contributed to several high-impact preclinical discovery programs, especially in infectious diseases, oncology, and nanomedicine. Today, Dr. Geda will be sharing his insights on recent innovations in reproductive nanomedicine, focusing on diagnosis and treatment. We are privileged to have him here with us. Now I will welcome Dr. Geda to the stage.
Now Dr. Uday Saxena come forward and felicitate our speaker, Dr. Gangaraju Geda.
Okay. Oh, you are asking to take it. Thank you. So I can control. Good morning to all. Can you hear me? I hope most of the people are new to my topic. Anyone have idea about the reproductive? Anyone is working? Okay, I guess it is also new for me. I have one request. Kindly don't take any photos. Okay, because some of the things I should not disclose. Even because of seeing all this esteemed conference, I have incorporated some data. And also I can't discuss few of the things, but still I consider, because my team told don't disclose. I hope all of you are very professional, so kindly don't take any pictures. The title is Recent Innovations in Reproductive Nanomedicine, Diagnosis and Treatment. I am not completely giving the details of the reproductive diagnosis and treatment. So what I am going to trying to do in this section is, as a pharmacy or chemistry, we mostly look at in the drugs development, medicine development, discovery. as well as drug delivery or antibiotic delivery towards microbial treatment or cancer treatment, Alzheimer treatment, some XYZ. So, the basic concept between the delivery of the drugs, any disease is similar. So, I am connecting this point towards the reproductive, how we can succeed. So, this is also very challenging and new, not only to me, throughout the world. There are many development in the reproductive system, but it is still very initial stage in the nanomedicine. The development of nanomedicine is very vast, and currently it is applicable in many areas, but it is very starting stage in the reproductive system because of the toxicology. So, I am going to cover as much as I can do, as well as I will give the information as well as much as I can. Yeah. Okay. Yeah. Don't. So, let us start the content. So, the conference, the main aim is integrative trends in pharmaceuticals and health science. So far all the sections we have listened from the experts from throughout the world about the discovery of the medicine, delivery of the medicine for the human diseases. So, the development of the medicines. But today what I am going, the human have to born, right? The reproductive means it is the newborn, reproduction. So, that is I am going to talk. Okay? It is not completely new. As of now, we still focus on human diseases. I am going to give some basic information about the reproductive system. The reproductive is nothing but production, reproduction. So, we have covered that. Our topic is conference content is pharmaceuticals and health science. So, we have covered that one, that all the drugs are developing for the health science or health, human health. So, the human production is, it is very well known. So, the male and female, the sperm and ovum, they will combine together, we will get the embryo. But currently, if you look at the reproduction system, the worldwide, the birth rate is decreasing. So, because there are the various factors for the decreasing the birth rate. So, mainly diet, imbalance of the diet and nutrition and smoking. Another major issue for the most of the diseases is the mental stress. So, the stress is cause for all the diseases. Another major problem is that environmental factors. Environmental factors means toxic chemicals, especially the plastic is very dangerous to the human reproduction, because there are some chemicals they use to development of the plastic by using synthetic chemicals. There is some plasticizer like bisphenol. If you look at the most of the plastic container, they will mention that BPA free or not. If they were the well quality products, they will include BPA free. So, the BPA free or some other chemicals, what will happen? So, when we are heating containers or especially storing the liquids, the BPA will release slowly. So, in the milk or water if we consume, that will enter into our human body. So, the plastic is very dangerous compared to all other factors, and also weight and physical interactions and sleep and other things. So, how we can improve the reproductive system, the birth rate? The main thing is that physical activity, increasing the immune system, and nutritional food, having the nutritional food, this is the most important thing, balancing the food. and also stop smoking and weight balancing and avoiding the exposure of the chemicals. These are the basic minimum things we have to follow to improve the fertility of either male or female. So what will happen, I have mentioned that in the previously, the food intake is very important. So the environmental factor, whatever I mentioned that this plastic or some other anything, if any toxic chemicals enter into our body, there is a two factor will happen. One is that oxidation stress, another one is that inflammation. The oxidation stress is the most dangerous thing towards the reproductive system. So oxidation stress is nothing but imbalance of the production of the ROS, the OH free radicals, O2 free radicals. These free radicals will damage the increase the oxidation stress, and it leads to the infertility. So balancing that free radicals, we have to make it neutralize. For that we require the antioxidants. These antioxidants can improve the fertility rate by controlling the or scavenging the free radicals. So this most of the current research is focusing on this area for the reproduction, for finding the new antioxidants to control the oxidation stress, as well as there is other kind of the research is going on controlling the anti-inflammatory. So if we look at the publications in the between the nutrition, it means that antioxidants, the work is focus is gradually increasing. It is initially starting from the 2000. There is also some other publications before the 2000, but later the people are focusing this direction, because most of the researchers, they don't focus on reproduction system, but still many research groups, they are focusing towards the reproductive system. So I am going to connect between this reproductive system and the nanotechnology. So there is a big gap between the nanotechnology and reproductive system. So as we know, the nanotechnology is not new. It has started 2000 years back, and still there are the many inventions are going on towards the nanotechnology in many various fields. So now presently research is also focusing on this reproductive system. So let us see the nanotechnology currently. They have applied in the information technology, various applications, and the energy, battery, solar cells, so on, and medicinals, cancer treatment, bone treatment, drug delivery, whatever all the sections we have covered on the medicine point, and also consumer goods. So nanotechnology have applied in every field. So in the biomedical application, if you going for the specifically, this nanotechnology applicable mainly focusing on treatments and diagnosis and targeting the diseases. The nanotechnology will have to targeting the diseases as well treatment and diagnosis. So in the reproductive system, there are various kinds of the materials already people are looking, like especially the liposomes. The liposome shows lower toxic compared to other materials. There are polymeric nanomaterials, silica and gold, solid lipid nanomaterials. I think the pharma people mostly they are focusing on this solid lipid nanoparticles and carbon materials and quantum dots. So I am also going to introduce one quantum dot today in our topic, but most of the quantum dots previously they made from the cadmium based or lead based and tellurium based, mercury based. So the researchers have found that those are very highly toxic. Even most of the antibacterial studies, if you are working on the nano, we will focusing on silver nanomaterials. But the people researchers are find silver nanoparticle even 15 microgram, it's also very toxic. So most of all the metal nanoparticles are rejected. If you look about when I started this project or 1.5 years back, I am also very new to this one. I have started to work on what are the nanoparticles are developed in the reproductive system. All the researchers, even if it is published in the high-end publications, they only focus on the toxicology of the nanomaterial, There is no development towards the diagnosis and treatment. There is a very little bit literature is available because of this toxicology. So most all the metal-based nanomaterials are not suitable for this reproductive system, but experimentally we have observed and found that liposomes are better, and the quantum dots, non-metal based, there are some quantum dots. We have focusing in that area. I am also talking about today's section. I will also give some information. So reproductive system is that there is the two-way infertility, and also there are the reproductive related many disorders. So the reproductive system is very complicated subject. Even especially the women have lot of the cancers. Itself, those cancers also related to this fertility rate. So there is as I mentioned earlier, there are different kinds of the materials. So when we are looking the nanomaterial for the any treatment or diagnosis, these functional groups will play major role. So when we are going for the treatment, we have to conjugate with the drugs, antibodies, or targeting agents, or imaging agents. So we have to look at the mechanism how to functionalize. So these functional groups are very important to conjugate between the drugs and nanomaterials, and we can make the nanomaterials different size and shapes, and also porosity. For loading the drugs, the surface area is very important. The porous material can load the drugs more than the non-porous materials, and we can also produce the materials various compositions. So these are the various kind of the materials. So for focusing on the reproductive systems. So as I mentioned earlier, I am going to talk about one candidate, that carbon dot. So the carbon dot in the biomedical application, I have started this carbon dot to work in the bioimaging for cancer and treatment like MRI, fluorescence imaging, and drug delivery, photothermal therapy, photodynamic therapy. The same thing I am forwarding towards the applying the reproductive system. So the beauty of this thing is that we can produce the carbon dots from the natural source like plants, leaves, fruits, fruit peel, all the things which I mentioned earlier. The diet is very important. So having the balanced antioxidants. So most of the leaves or fruits or fruit peel having the antioxidants. So we are producing the carbon dots from those things. It means that we are converting natural sources into nanomaterials. So there is no non-metal nanomaterials. So we have found that even though this material also shows the toxicity, but compared to metal nanoparticles, they show better biocompatibility. So we have plenty of precursors to produce the carbon dots from the vegetables, fruits, waste material, food, and leaves, all the things. I am not going more details about these things. So in our research, we have prepared the carbon dots from the papaya and neem and orange peels. All these works are already published. So here there are some other applications, but the main application is that the carbon dot shows that good antioxidant activity. So ascorbic acid, as we know, it is a very standard antioxidant agent to compare anything. That is standard. So when we have prepared the carbon dots from these natural sources, we also observe that this carbon dot shows very good antioxidants, and they also show the fluorescence. That fluorescence also we applied in the imaging of the reproductive cells. So this is the confirmation of the antioxidant activity. So we have confirmed that the materials as showed. This is very simple DPPH assay we have used. So for confirmation of the antioxidant, before going to cell work, we better to perform this one to find the antioxidant activity of the materials. So this is the major point any biomedical, as we know, the toxicology study. So as I mentioned earlier, all our materials, we also have assessed with some metal nanomaterials like silver. Even the lower concentrate, the silver shows very toxic. Some other metal nanoparticles we have assessed. Finally, we concluded that this carbon dot shows that for the animal, the birth of the, here is, we didn't check the specific the reproductive cells. Initially we have assessed the toxicity for the zebrafish completely from the egg to formation of the zebrafish. So here we didn't find any much toxic for the formation of the zebrafish from egg to fish. So later we have moved towards the study the reproductive specific cells. One is the TM3 lignite cells and another one is TM4. So up to 500 microgram we are able to manage the toxicology. But when it is going to other nanomaterial, even 50 microgram also we are finding the toxicity. So finally we concluded that these carbon dots we want to invest more toward this toxicology studies. So then we have also compared with the normal cells. So when we compared with the normal cells, even 1 mg also the carbon dot doesn't show any toxic. But when it is coming to the reproductive cells, up to 500 grams we can consider, because when we are going to look at the bioimaging of this carbon dot shows the fluorescence itself, we no need to use any dyes. So we are unable to see the imaging at 200 microgram, 100 microgram. So we are optimizing the concentration and considering 500 microgram. So initially we have checked with that carbon dot with the normal cells. So here we have control and carbon dots and carbon dot with folic acid, because why we have used folic acid? Folic acid is the targeting agent for the many cells receptors. So then, so we also have developed other material magnetic carbon dots for the bio MRI imaging and fluorescence imaging. And also we have assessed the carbon dots for the bioimaging, because before I have mentioned that they have shown the fluorescence. So we have used the imaging of the zebrafish. So it means that the carbon dot can replace the all organic dyes or some quantum dots. Previously, before coming to the carbon dot in the picture, scientists usually focus on the metal-based, cadmium-based quantum dots. So these carbon dots also replace the metal-based quantum dots. So this is the reproductive cells, TM3. As I mentioned earlier, so we can able to see the imaging of the cell from the 200 microgram. But in the 500 microgram, we are able to see, but still the toxicology is showing, but compared to other materials, this candidate is better. So even the TM4 also we have found the similar strategy. So we also use the sperm to imaging. So the carbon dots are internalized very well with the sperm. So here we have functionalized the carbon dot with targeting agents to reach to the sperm. So we can see that the imaging, the full sperm we can find. This is the male spermatozoa. So now I am moving towards the drug delivery. Unfortunately, I can't give more data information here, but I can give the strategy, the approach, the methodology, how we have developed. So as we know, all the sections covered the drug delivery. If we take the drug alone, there are the many problems. So one biggest problem is that reaching the targeting. Another one is that we require the high amount of the drug. So and also another one is solubility. So the nanotechnology resolve the all traditional drug delivery limitations. So carbon dots in the drug delivery. So reaching the target is very important. So for the targeting, we usually use the targeting agents to reach to the receptors. So the nanotechnology can help to integrate with these targeting agents and also drug moiety. Drug moiety means doxorubicin, sRNA, so on, so whatever. So we can also incorporate the imaging agent in this nano system. So nano system, it basically deliver the imaging agents or therapeutic agents for the delivery system. So here, this is the example for the cancer. We have used the carbon dots functionalized with the folic acid, that is the targeting agent, and doxorubicin. We have found lower pH. The material has delivered the drugs very well. So the same strategy we are going to apply in the reproductive system. So any drug delivery, the 3 points are common. One is that nanomaterial to deliver the targeting agents or imaging agents towards the targeting agent. Another one is that targeting agent, nano targeting agent and therapeutic agent for the treatment. So these 3 combinations are common for any kind of any disease drug delivery system. But we have to find that which targeting agent, which therapeutic agent, which nanomaterials. This selection is the most important criteria. So when we are looking for the drug delivery system, we have found that the liposomes are better drug delivery systems. They have the hydrophobic and hydrophilic natures. So we can incorporate drug inside the liposomes, or we can conjugate. So they have, in our experiments, we have found that these liposomes are very good material for the deliver these reproductive therapeutic agents. So basically, what will be happening in here? The first step is drug delivery vehicle. So the drug delivery vehicle, here we have used that lipid nanoparticle. So encapsulating sRNA and BSA. So sRNA, from the literature we have found that that is improving the therapeutic agents. We are also incorporating antioxidants into this nano systems. Though why we are incorporating antioxidants means that antioxidants can be maintained decreasing the oxidation stress. So the main point is step is that developing the drug delivery vehicle means selection of the nanomaterial, preparation of the nanomaterial, characterization of nanomaterial is the first step in the development. Next one, payload of the therapeutic agent. We have used sRNA therapeutic agent for the delivery. Second thing is that we have found the more challenging finding the receptors in the reproductive cells and the targeting agents. So after many trials, we found that BSA is the better targeting agent towards the reproductive cells. So we have conjugated this BSA into the nano system along with the sRNA and also some other antioxidants. Then we have to deliver these things to spermatozoa. So there we have found that the sRNA release, and we observed that the cell death rate is less. So here basically what we are doing that for the reproductive cells, we are making interaction with some toxic chemicals. So the toxic chemicals are increasing the oxidation stress. When we are giving this antioxidant, those are the decreasing the oxidation stress. So it will be increasing we found that the fertility rate is increasing in this system. So this is some experimental basic results. For the conjugation of the thing, we have found that fluorescence. So we also see the fluorescence of these compounds, and also some other confirmation for this one. So we already, this work is ongoing. Unfortunately, I can't show more than this. I have limitation to show the other details, but I hope I gave the concept applying the drug delivery system in the cancer to the reproductive system, imaging from the cancer to the reproductive system. We are applying the same method what we are doing the cancer, some other treatments. So we are adapting same method towards the reproductive system. So in the conclusion, the development of nanomedicine has opened new horizon. So when we are applying the grants in India, most of the times we apply the mostly cancer. Most of the projects are rejecting. because everyone is focusing on the cancer, they are not focusing on other area. Of course, they are focusing on other area, but this is another new area. There is a lot of scope to explore the new things, and it's a very important thing for the development of nanomedicine towards the diagnosis, treatment, and prevention of the reproductive diseases. This transformative approach from the nano to the reproductive system conditions such as infertility, endometriosis, and reproductive cancers. So we can develop the new nanomedicine to improve the reproductive system. I hope some of the people can focus on this area. If anyone is interested towards this research, you people can approach me, we can have work together. So this is about my research laboratory. So we have working on the endocrinology, reproductive biology. So we mostly focusing on the toxic chemicals impact on the reproductive system and how to control this oxidation stress. So this is my group. Currently we are working in South Korea. And these are my funding agencies, NRF from South Korea, DST and UCSI from Malaysia. And also there are many collaborations throughout the world. So today I am here because of all these collaborations. So in the collaboration, research is very important in the science. That's what I found it. So thank you to all. If you have any question, you can ask.
Thank you very much, Dr. Geda. Very interesting presentation. Are there any questions, please?
Hello. Yeah, a very interesting talk. So you have showed that zebrafish toxicity study, and but it's clear that this even 50 microgram dose nanoparticle, they have cardiac problem. You see the pericardial edema.
That's what I'm saying. This is, we are facing the problem towards the toxicology when we are going with the specific reproductive cells. In the zebrafish, we are checking with the toxic throughout the body. But when we check the specific cells, we are facing the problem there.
Even zebrafish are the toxic. It is showing that they are toxic to the fish because the the heart, they have the pericardial edema. Okay. It's clear that they are toxic even at the 50 microgram.
Okay, okay.
And also you can see that also you have shown that nanoparticle is basically incorporated in the fish.
That zebrafish study is from the egg to formation of the fish. So we are assessing the fish is forming or not. Fish is not completely die there.
Okay. But it is toxic, okay.
Yeah, it's toxic, but the fish is not die. But when we are checking other materials, fish is not growing from the egg to formation.
Okay, okay. Fine, thank you.
Okay, thank you.
Any other questions for Dr. Geda? Yeah, please. No? Okay. Any questions from the students? Okay. Thank you, Dr. Geda. Very nice presentation again. Yeah, thanks a lot. Yeah. So move on to the next speaker. Yeah. Our next speaker that I would like to introduce is Chinmoy Patra. He is a professor, scientist at Agarkar Research Institute, Pune, with a distinguished academic background. He earned an All India rank 1 in GATE 2005, completed his MTech at IIT, and pursued a DAD fellowship at TU Dresden. Dr. Patra's doctoral research at the Max Planck Institute focused on cardiac valve development in zebrafish. His postdoctoral work resulted in several high-impact publications. In 2014, he established his lab at Agarkar Research Institute, specializing in cardiovascular development and heart regeneration using zebrafish models. His research has been featured in top journals like Science and Cell and Bioscience. He has received numerous awards, including the Max Planck Partner Group Award and DBT Wellcome Trust India Alliance. Today, he will talk about zebrafish disease models to study human diseases. exploring their role in advancing disease research and testing. Please join me, and it's my pleasure to invite you to present. Yeah.
Thank you very much, sir, for a
So
nice introduction.
now Dr. Uday Saxena will come forward and felicitate Dr. Chinmay Patra.
Thank you, sir. Yeah. Yeah, thank you. Thanks. Fine, let's start. So, thank you very much, sir, for your kind introduction. And I'm very, I should be thankful to the organizers for inviting me to share our research, what we are doing last 10 years in my lab at Agarkar Institute, Pune. And thank you all for listening me. And my expectation that you please, develop questions when I present something. So that way we can do a little bit of introductions. I'll try to finish in 35 minutes so we can have 5 minutes for discussion. So sir, please also remind me that when I'm done around 30 minutes. Perfect. Okay, so what I'm going to do today that we in our lab at Agarkar Research Institute, we use zebrafish as a model organism to study mostly cardiovascular biology, the heart development and also heart regeneration. So, I'm audible? Am I? Yeah, fine, good. So, oh. Go back and this is full screen there. Yes. So whenever we think about any kind of research, ultimately start with the disease. And if you basically look at the disease, here I separated into two forms. One is the disease that are not genetically linked. Basically, we have during our day-to-day life. It can be infection, it can be some, hyperacidity or kind of, it can be the fever, or it can be the extreme like the cardiac problem or any kind of organ problem. But there are also class of disease that basically we carry when we born. So this disease sometimes you can see by facial expression, you can see that we have some problem. But believe me, in this group also, people some of having some genetic problem or some genes are basically mutated or some abnormal these things are there, but we are not seeing it. So probably that is doing some problem inside our body, and sometimes it develops. Like in some cases, like at the age of 30 or 60 or 50, some kind of disease develop, and that are basically due to the mutation, the genetic mutation. So these two class, whatever this class, whether it is in general day-to-day life or due to the genetic abnormality, the disease form, and if we want to repair this disease, we have to find a solution. And to know the solution, what we need to know, that where the problem. And in our lab, we basically try to find out that how the disease is developing or where the problem, using zebrafish as a model organism. So this our lab use that, but if you look in general that what throughout the globe what we are doing, we mostly use disease animal model because we can't use human as a experimental model system. And what is come in our mind that basically that rodents and also like primates, and it can be also guinea pigs and other animals. But if you look at that history, that people use mostly other organisms. So many the research development happened through using this organism. It starts from like around 2,500 years ago using the sponge. That is how the sponge basically came into the research is like around 2,500 years from now. In the Greek people, they are along the seaside, so they have a lot of sponge, and they found that if the sponge is destroyed, that then every sponge piece they make new sponge. So that's why they thought the extract of this sponge will be helpful for the regeneration of human. This skin. If there is a skin cut, so if you put the extract of the sponge, then may also help in the healing. So this way, people used to use the sponge in regenerative biology. In case of the zebrafish, it is recently, I would say, in last 4 or 5 decades, we are using zebrafish as a model organism for in different field of research. This started in USA, but if you look at the fish, they are mostly from our country or maybe neighboring country like Nepal, Bhutan, Myanmar in this area. How it moved to the US? Basically, these are the pet animal. So these are all modified zebrafish. You can see it's colorful animals. They are you will find them throughout the globe, and also in the Indian pet shop you can find them. But in the lab, we use this wild type fish like gray in color. So how we maintain that? Because we need to maintain that. So in the lab, whoever basically use zebrafish as a model organism, we maintain them in very individual system. So you can you please switch off these stage lights? So you can see that here the 2.5 to 3 centimeter long fish, these are the adult fish. But if you look at here also, we are growing around 15 days old fish, you can see over here. So you can see this all this. So basically this we grow everything, and then we keep individual tanks and individual fish. And I'm showing here also, you can come down. So these are actually the breeding tank. We can keep male and female over there, and you can get the eggs collected over there. So if you think about the research, what we do, the relatively most simple form of the research we use in the lab, it is the in vitro cell culture. Yeah, I am telling that relatively simple because it's also not easy to do it. But in this case, what we can see, how cells are proliferating, how cells are migrating, everything we can observe under the microscope. But this is not recapitulating our body, because this is the only one type of cells. Our body is not made up of only one type of cells or system. So that's why that people now are using organoids. Here people put like 2 to 3 different type of cells. But again, this is not enough because it does not have any other control like neuronal control, blood vasculature, lymphatics, nothing is there. So that is not also enough. So we have to move to toxicological study and even pharmacological study to the rodents and then primates and human. So I just make it like that. It is a long process. It's just like making a road around the hilly areas. It's long, and it can be shorter by making a tunnel. So you can make it shorter, and I put zebrafish as in that you can make this long distance travel into the short distance using zebrafish. Why I am telling? So you can see that this is the in vitro system where we can see everything is happening, but we need in vivo, means whole organism that is there. But zebrafish, I would say this is a in vivo in vitro system, in vitro in vivo system, because if, as I have shown earlier, if you keep a male and female together, you can get these fertilized eggs next morning. So you can see, and this eggs looks like this. So our life start with a single cell. Similarly, their life also start with this single cell. Within 30 hours, you can see nicely formed zebrafish. This heart is beating. In human, it takes 3 weeks to beat the heart. For the zebrafish, it takes 21 hours to beat the heart. So it's very fast. You can see this is the yolk sac, particularly food reservoir. So first 5 days they don't eat. So they get all the food from this yolk sac, and after 5 days they go through metamorphosis and they don't have any yolk sac. They start to eat, and all these metabolism happen as it regular happen. They are pretty transparent. You can see here I am showing a 4 days old fish. They are all the blood vessels. So you can see every blood vessel. We can see every cell, even how moving in. I am going to show you a couple of movie that how the cells are moving inside the blood vessels. They are high fecundity, means every fish, basically every female lay around 100 to 300 eggs per week. So you don't need to kill anything. You can get easily these eggs every week, 100 to 300 from one. They have efficient regenerative capacity. They can regenerate each and every organ. For our case, we regenerate like skin or maybe liver, but they can regenerate brain, heart, kidney, all organs they can regenerate. And we can make also easily the transgenic lines. So as I told, that very transparent they are. So you can see this is the 2 days old fish. They have the beating heart over there. So I am showing a, this is the scanned heart from this fish. They do have 2 chamber heart, not like us, 4 chamber, 1 atrium, 1 ventricle, and you can see every green cells is basically cardiomyocyte. There are the contractile cells, they contract. So our heart contract, that is responsible for, cardiomyocytes are the responsible for this contractility. So they do have the, this cardiomyocytes, and you can see that is the optical sections through the heart. This is from double transgenic animal, where basically greens are indicating cardiomyocytes or myocardial layer, and 1 endocardial layer, you can see the lining. So similarly, like also in human, when the heart form, they have only 2 layer of cells. So they do have the 2 layer of cells, and in between you can see the free space, that is basically jelly-like structure. There is no cells. This cell-free layer is called cardiac jelly. So what I am showing, this is exactly also happen in case of the human. So you can see here the ventricle and atrium 2 chamber, and in between the canal over here, there is no valve. So basically initially valve yet to form over here, but if you look after 10 days, this is 2 days, after you can see 10 days old, the same fish, same heart, you can see the valve triplet over there, you can see the complex structure over here. These is finger-like projections, they grow inside. So they are very important, these structures, these are called cardiac trabeculae. So why they are important? When the atrial pump the blood, it just hit the tip of this trabeculae, and that sends to the SA node that give the next signal. So that's why heart basically contract. And 2/3 of the contractile force come from these trabecular cells. And human disease called cardiac non-compaction, that is also because of the trabeculation defect in human. So as I told that, you can also see that how heart is functioning over there. So this is very, that you don't need any hi-fi microscopy. This is under the stereo microscope, you can see that how the every blood cells is moving from the atrium, going through the atrioventricular canal, going to the ventricle. You can see that now it is muscular structure because it's 4 days old, and then you going through the outflow tract, and this is the gills, because their gills are the oxygenating center, like lungs we do have, and from the gills it basically go through the, through the body and come back to the atrium over here. So we can, with a high speed camera, we can even count how many blood cells are moving through this atrioventricular canal, and you can see here that is basically the valve forming area. You can see that is basically trying to close it. If you want to look at the cellular level without killing any animal, you can see even the contractility force of the heart. So here basically I am showing a heart where every this gray color or white you are seeing here, F-actin or filamentous actin, and you can see that how big particularly these cardiomyocytes, how big it enlarging and is coming back, and these are the nuclei inside the cardiomyocytes. So basically you should know that also in our case that when this cardiomyocyte contract, that basically the length of this come to the almost 5 to 8 times shorter. So one can measure that shortening of this cardiomyocyte length during the systolic and diastolic condition. So is all these beautiful, but then come to the point how it will be beneficial for human. So of course the similarity is important things. So around 70% of the genes are similar between the human and zebrafish, and that is true for everything. If you look at the rats or mice and with the human, only 70% genes are similar. Rest 30% are species specific, because that you can say that the mouse having the tails, we don't have it. So this because of all these different characteristics of our body characteristics, the 30% genes are responsible. So those are basically species specific genes. So now that around 13,000 protein coding genes out of 20,000 are similar between these two organisms, human and zebrafish, and zebrafish carry all the organs except lungs. So that means the function of these common genes can during the development of an organism or in any kind of pathological situation can be studied using the zebrafish as a model organism. And that's why nowadays if you look that we are basically people are using zebrafish in biomedical research in different area. Just only 5 I am giving the example. One is modeling of human diseases. I will mostly talk about the modeling of human diseases. Then one can, if you have the model for the human disease, then you can take this model for the drug screening, that which molecule will be beneficial in any particular disease. That can be done. And nowadays is very popular, the two terminology you can see. One is personalized medicine. So why personalized medicine? Because you can make a disease model in zebrafish having the similar problem what having this particular patient. that can be done, and drug repurposing. So one can also do the high throughput screening by modeling of human diseases in zebrafish and go for high throughput screening to find out get the new purpose of the drug. Of course, similarly toxicity study one can do, and the another important area is the regenerative medicine, because zebrafish can regenerate each and every organ. So we can understand how naturally they can regenerate the organ, and if we know the process, we can extrapolate to human. So I'll start with a molecule is called actin binding protein 1, and actin binding protein is nothing, for simple that I show you the beating heart having this white color structure, that is basically F-actin. And this actin binding protein is developed, expressed, and basically bind to the tip of this gray color structure I have shown in the beating heart and maintain the length of F-actin, whether it will be longer or shorter. So it does like that, and the paper came out and showed that in human patient, this in Europe, that they have found 3 patients from 2 different family, and these 3 patients having the mutation in that particular gene, and all the patients what they have, this is the basically human heart model. You can see this is the ventricle, at 2 ventricles we do have, and these are the atrium. So what these all the 3 patients having, they have these atriums are basically enlarged. So they have normal left ventricle, but they do have bilateral dilatation. So that basically they get enlarged, and they do have valve problem. So these 2 problems are there, but the beautiful thing is over there, these patients, they don't develop or they show any problem till 9 to 10 years of age, as I told. So they are all looking morphological normal everything. They basically complain that they have migraine when they develop 10 or 11 years old, and then visited the doctor, and doctor basically make this test and everything, and then ultrasound, they found that they have the cardiac problem. And then basically they sequenced and identified that the gene is basically muted. It's fine. And in case of the mouse, as soon as this gene has been shown that the mutated or knockout, mouse basically die in utero. So they basically don't born. But then we thought, okay, why not to look at the zebrafish, because the heartbeat in zebrafish and zebrafish and human is pretty similar, closer to 100 beats per minute. So we looked at first that whether this gene at all expressed in zebrafish. Yes, indeed, it expressed in the zebrafish heart. You can see this is the blue is indicating the expression of the gene, and it also expressed in the adult heart that we have seen here over here. And then we have introduced a mutation within the CRISPR-Cas in this, and then look at this animal what is happening. So if you look at the 5 days, they don't have any problem. This is basically the heart is here. This is the pericardial cavity. You can see there is no problem. But if you keep this fish up to 4 months, you can see nice pericardial edema, which indicating they have the cardiac problem. And exactly happen in the human that at the age of 9 or 10, they have shown that cardiac problem, the fish having also the problem over there. And when we isolated the heart, you can see this is the atrium, this is the outflow tract, and this is the ventricle. If you look at the mutant, the atrium, huge atrium, and you see the atrium is almost like 5 times enlarged in compared to the atrium of the wild type. So it's matching, and then you can see that this is the ventricle size almost similar over there. So basically shown that similar to human, that they do have problem in that case. So that now that we know that, okay, now we have developed a model to study that human ABP1 mutation in zebrafish. So now we know that only that is matching. The next what is happening and what is the look, this at the tissue. So this is the atrium, you can see, again you can see the atrium is such a big, and this is the ventricle, relatively similar. If you look interestingly over here, this is the wild type scenario. If you look at this trabeculae structure, here much more and much thinner. I think you appreciate that these trabeculae are much thinner in compared to the wild type situation. So we found that basically they do have trabeculation problem in this adult fish, and we are actually studying that how this is developing. So we are studying from the very early stage. Then as has been shown in the human, they do have the valve problem. So you look at the cardiac valve by using the collagen staining. So I have isolated the heart and stained for the collagen. So here you can see the structure, collagen positive, this structure is basically the valve. You can see over there. So this is the wild type valve, you can see the structure, and you see in the case of the collagen positive, the valve is much smaller. You can see over there, this structure is much smaller. So we do now see that zebrafish now ABP1 mutant can be used to study the similar problem having in the human. Now come to the next part, that is if you have like some disease model or anything, what we can do the drug screening. So when you think about the drug screening, it can be done anything. So being a pharmacist, there are 4 important things to need to know. If you consume a drug, or if we get a drug by testing zebrafish, whether it will be useful in human, that is important, that targets. Because zebrafish might have the targets, it beautifully work, but we don't have, this drug does not have target in human, so it will not work. But it's already studied has on around 90% targets for the existing small molecules are conserved between human and zebrafish. So we can be sure that 90% there is a chance if we find something in zebrafish can be useful in case of the human. Another is the metabolism. So you know that also that when you consume a drug, body will take it as a foreign material. So body has to metabolize it and then to make it simplified to get it excreted out. So the rate of metabolism also important, because if a molecule, this metabolism is different between these 2 organisms, then we cannot also use. Because if the metabolism rate is higher over here than the human, then the molecule will not be shown toxin to this, but it will be toxic to the human. Other way around, if the metabolism is too fast over here, so we will not see the pharmacological effect, because this is immediately it getting metabolized and out, so we cannot. So that's why it also important that metabolism process should be similar, and indeed also around 90% similarity present in the metabolism system between the human and zebrafish. So we can use it. For the heart or cardiovascular system, with this most important thing, the ECG or the electrocardiography of what we look. So if you look at the mouse or rodent, it can be mouse, rat, guinea pig. So these all the rodents having high, the cardiac contraction frequency, there you can see around 500 to 600, whether zebrafish have human having quite similar, you can see over there around 80 to 100 beats per minute. So that means also it is pretty conserved. So we can use the zebrafish as a model organism to study the for the drug screening or the toxicity study. Now I'll give a small the example, one small project from our lab. So if you look at, we are looking at that tumor development or the solid tumors for the cancer. That study, if you look at the tumor development, so it's basically when tumor developed, a lump of cells basically grow, but there is no blood vessels. Because this is not natural process, so few cells basically get mutated and proliferated, make a heap of cells without any blood vessels. And when there is no blood vessels, this is good for the body because the cell will die. There is no blood vessels, no food, no oxygen, anything. But what this tumor cell does, it secretes some molecule, this is the chemoattractant, and they then basically send this to the nearby blood vessels, and they start to make new vessels. This is because of the hypoxic situation. So whatever this secreting, this is because of the hypoxia, because there is no oxygen, and this secretion lead to formation of the new blood vessels. And when new blood vessels come inside, they get lot of food and everything, and they slowly grow more and become metastasis. So then grow to other part of the body. So that when anti-cancer for the solid tumor, people look two area. One is killing those cells, another is inhibiting the new blood vessel formation. So keeping this thing in mind, we look at the zebrafish embryo. As I mentioned that till 5 days they don't have everything. So that means early development of these blood vessels during like first day of their life, they don't have proper oxygen amount what is required. So that is they are in the hypoxic situation. So this in this hypoxic situation, if you look this particular blood vessel cells, so this you can see the few cells trying to make blood vessels, and in 50 hours they make a basket of blood vessels. You can see the network. So this process happening is relatively in hypoxic situation. Considering that, we can use this to study that which molecule can inhibit this process to find out anti-angiogenic drug. So with the in collaboration with the TIFR Mumbai, we study this kind of processes. So what we do here, we take like use this 96 well plate and take wild type fish expressing this green fluorescence protein in blood vessels and keep 4 to 5 emiros in each well. So in this 96 well plate, if you take one positive control and one negative control, you can still having like around 90 molecule you can test at a time. So you can put 90 molecules in every individual tanks and look up the processes. So what we used to do, we used to take 30 hours post fertilized animal over here and put the chemicals whatever is our chemical of interest and look after 50 hours. and then after 50 hours we looked at whether the growth is like this is the normal, or whether there is a inhibition, or sometimes you should see also more number of blood vessels. So with this what we found, one is the ellagic acid. So what we observed that in the control you can see nicely grown that basket what I have shown over here, and this is basically the area I am showing, and you can see that ellagic acid inhibit this process, and that is also dose dependent. As we increase the dose is basically look at that, and also we looked at that as what dose basically it shows any kind of morphological defects, and we found that up to 400 to 800 micromolar they don't have any morphological defect, but one can see that 100 micromolar is enough to inhibit the blood vessel. Now come to my last part. How long do I have time? 10 minutes? Perfect. So this is the last part of my talk. This is the hardcore area we basically in our lab we do. So why you are certainly interested in the heart regeneration? If you look at the myocardial infarction or heart attack, that's basically nowadays it's like not 15 million, it's old data. Now 20 million people die every year due to the myocardial infarction throughout the globe. What happened in myocardial infarction is start with blockage of coronary vessels. So what I am showing here, the blood vessels or coronary vessels inside a zebrafish heart, the adult heart. So adult zebrafish heart, you can see this all blood vessels. So if you imagine that there is a blockage over here, so then this part of the tissue will not get the oxygenated blood or any kind of the food. So if they does not get oxygen, what happen? It go through the cyanosis. So these cells over there they die, correct? Because you see that when the heart contract is, they need lot of energy, lot of oxygen they don't need. So as soon as the blockage happen over there, then you see the cyanosis, and if you cut that, you see that this kind of structure. This is nothing, this is basically the collagenous scar. So when the cells or cardiomyocyte they do die, this part has to be sealed, because body does not want that heart get rupture. So if there is no cell survive, so heart will rupture and patient will die. So body what does? It makes a lot of extracellular matrix to secrete it out and seal that, make an wall. But this is not contractile place. So that what happened in this scenario, basically this part of the heart beat, but this part remains silent. But this part also does not remain silent, heart become arrhythmic. Why they become arrhythmic? You can see that some tissue, the cardiac muscle tissue present inside over here like an island. So when the heart beat, whole heart will beat, but this is not connected to this one, and these island beat separately, and which lead to cardiac arrhythmia, and that is most dangerous in case of the patient when they do have this lot of this the island of the survived cardiomyocyte. So this is there, and people believing that scar is a problem. But when I started my lab in 2014, I actually saw the interest to, before that I was not working in the heart regeneration. So I thought, okay, look at the zebrafish heart regeneration, what we know. So in zebrafish heart regeneration, what we published paper at that time showed that they also goes through the scar tissue formation, but they can completely regenerate. So I basically hypothesized that the scar whatever happening, that's not a problem, but the composition of the scar is a problem. So composition might be different in between these two scars. Those both having scar, but different composition, and that basically telling that whether heart will regenerate or heart will not regenerate. So okay, so that's why now my lab works basically try to find out that the scar forming molecules and their role in the heart regeneration. So when you study any kind of regeneration, first one need to develop a system where you can make some problem or make injury first. And what I am going to do that, okay, how we make injury in the zebrafish heart. So it's very simple. We take, you can have this fish into the sleeping solution, is Trican solution you keep. In 2 to 3 minutes the fish will sleep. These are simple, all the instruments you need. This is the sponge, it's also wet in this sleeping solution. Keeping this copper wire inside liquid nitrogen to make the super cold probe. And now the student is keeping the fish, anesthetized fish on the sponge. Now you can see this is the thoracic area, and he is removing basically scales from there and making a hole to get the heart access to the heart cardiac ventricle, because this part has to be injured, and that is next to the skin actually. So you see that now the beating, this ventricle, what you have seen basically this one. So you have the beating heart, you take out the super cold probe, this is the copper wire, you can put over there, and it burn the tip of the apex. it will be immediately stick. You know, that is like the burning, same thing. It's not hot burn, it is a cold burn. So you just put on the tip of the apex, and it will only the kill around 25 to 30%. That exactly happened in the heart attack, around 30% cell death in our case. And you keep it over there within 1 to 2 minutes. It basically you know, not start to swim, because it will also not feel good. One second. Sorry. So it is basically it's settle down and stay for, you know, for 2-3 days. It will not eat anything, and then slowly it start to come to you know, normal behavior. So now we look at that. Okay, we basically scan many extracellular matrix or the scar forming molecules. So here, for example, I am showing you that one molecule is called CCN2 or CTGF, and this is the heart after injury. 7 days after injury, cut it out and then make the sections through it. This is the healthy tissue. You can see the red, and this is the injured tissue, and green is indicating that the protein expression. So if you look at this green protein is expressed at the regenerating part of the tissue over here, means this part, and also present here. That part will be regenerated maybe in next 1 or 2 months. So that basically told us that probably this molecule help heart regeneration, because it is associated with the cardiac regeneration processes. So to understand this, whether this molecule is important for the heart regeneration, what we have done in the lab, we basically remove this gene from the fish. So we have removed by CRISPR-Cas technology. Now this one we have done with the TALEN technology. So we have removed these genes from the heart, and we make the injury, whether it is wild type and the mutant, and study there in the future. So after 60 days, we have isolated the heart, because we know that by 60 days their heart can regenerate. You can see this, the healthy heart, means wild type heart. We make injury over here. So you can see that part over there. This is indicating the cardiomyocyte, the red, and you see that part is uninjured and over there. So if you basically you can see that the cardiomyocyte, the structure of the cell, you know, you can see the much organized over there, but this part is not, which basically telling that is newly formed cardiac tissue, which need more time to be completely organized like the healthy tissue. So that's why we can see that, okay, which part is newly regenerated. But if you look at the mutant or the knockout of CTGF, where CTGF is not there, we remove the gene, so you can see the whole part is empty. There is no cardiac muscle. But the other part you can see, like here, very nicely organized the cardiac tissue. So which basically tell us that this gene, that CTGF, is very much important for the normal regeneration of the heart regeneration over there. If we look at here, we have isolated the heart. You can see that the beating heart of the wild type scenario over here, but in case of the mutant, when you can see the scar tissue. Exactly, you know, human heart after myocardial infarction, if you look, it will be looking like this if you look after 7 days. So this is the scar tissue uninjured. But what I was mentioning, that some, you know, this heart is beating, but here you can see that some spot that is beating, basically individual beating, that is arrhythmia. It's called arrhythmia because that is, those cardiomyocytes are not connected to the signal from the SA node, which is sitting over here. So the SA node only signal is reaching over here, then the scar, then the island is over there. So this is called also cardiac arrhythmia. So this problem is there. Since it is showing there is a scar, we look at the scar, and what we see that on the section, so you can see the collagen scar over there is almost gone in case of the wild type scenario, and but this is after 60 days, but after 5 months you see there is no scar, so all completely regenerated. But if you see that the mutant, you see the scar tissue huge present, you know, after 60 days, and it also present after, you know, 105 months. So it's basically, it's this saying that, okay, it is very important for the regeneration. So if you look at any kind of tissue regeneration, there are important thing is that first will happen the inflammation. So you have the skin cut, you see the oozing and all these things happen, and then the scarring. And if you don't want to see the scar mark on your face, then you need that blood vessel need to be grown, and then the cell proliferation, cell migrate. So you basically, all of us having the experience, if there is any cut mark on the face, that goes much faster than if anything having the finger. The reason is that our face having lot of blood capillaries. So blood supply more, so the scar removal, this process is much faster. So that means we need, when you look at the scar, we have to think about that whether this blood vessels growing properly or not, first thing, and second, the functional cells are coming and replacing the scar with the new tissue. So this one very brilliant student, this Devanjan Mukherjee, have worked on and this is basically discovery happened without knowing it. We did not plan for it. So how it is happened? So he was only looking at the blood vessels, that whether it's grown or not, but in those fish were double transgenic. So by chance he scanned and find one more interesting thing. So first he looked at the blood vessels, but he saw that in this double knockout that you can see the red is basically the cardiomyocyte nuclei. So all the contractile cells nuclei in the red. So he found that all the red cells migrated, and you can see that all are aligning with the blood vessels. So this way the discovery came. He has first time shown that this cardiomyocyte migrate along the blood vessels to repopulate this area. But then another finding is that in the case of the knockout or the mutant, you can see this is the knockout of CTGFA. So you see that nicely grown blood vessels is there, but there is no cells are coming on. So I told you, for the removal of the scar, you need the blood vessels, but that is not enough. You need the functional cardiomyocytes or functional cells to be migrated. So although in this knockout blood vessels there, the cells did not migrate. So the CCN2A is not required for the blood vessel formation, but it is required for this migration of the cells along the blood vessels. I am not showing all these data, but I am showing you here that massive phenotype you can see. After 12 days, you can see the migrating cells over here in the injured tissue, which is basically missing over here. So that is clearly telling that CCN2A, this protein is very important for the migration on regeneration of the heart. Next, I am jumped to that. What happens we give extra amount of this protein, because you know that protein is required for the heart regeneration. So if we give extra amount of this protein is like a drug, whether it can improve the regeneration. So
Sir, can I one thing that you were watching the
as per this, the function of the genes, the F-actin sarcomeric structure will be stabilized using this ABP1. So stabilized means we should see the longer filament in the mutated form, but it does not happen neither in the human samples and nor in our fish sample. So that is basically interest of our that, and we know now that this, because what is happening like that, is like, you know, you gave some work to somebody that, okay, please go do this work, and you know that is not going to happen, or your boss knows that, okay, I gave you that, like, poster work has to be done by you, and you are not coming, you are sick, so that means poster work will not happen. Yes, that is the case. But we found that the poster work, we have predicted that, okay, by you it will be done by 5 hours, but it happened like within an hour all posters done, because other 5 people were waiting. If you are not there, you are the barrier. You are the working as well as stopping not to come. As soon as you move, that 5 people enter and do the work very perfectly, not perfectly, because they have done more than required. So exactly this protein, when is not there, is nothing to do with the function of the protein. In the absence of or the lack of this protein function, some other protein come into the business and making more stabilized F-actin, which is not allowing heart to function properly, and that lead to overload of the heart and pushing the heart to enlarge. It's like balloon. You are pumping at more and then, you know, getting bigger and bigger. So this is very interesting. I'm not presenting everything because it's not not published. So we are just, you know, yeah.
Thank you.
But yeah, very interesting. Yeah. Thank you.
Any other questions? Go ahead.
I have just a curiosity. How long it can survive without what are these zebrafish? Because how much time it takes to rupture the heart and do the entire process?
Yeah, we can keep it 5 minutes. Even we
Okay.
can do the micro-CT keeping it 10 minutes outside the water, just wet sponge. So 10 minutes they are fine. Then we come bring back and put into normal water. Yeah.
Okay, okay, thank you.
More question there.
A good presentation. This is Dr. Harishankar. It's a good alternative animal model, fine. But how we are coming to the carcinogenic studies, how we are going to measure the tumor volume or disease progression or regression?
So we are doing, we have started just, I will not tell that, okay, we are doing, because there are many labs in the world that are doing xenografting. So we can take the human patient cells and grow it into the fish, and you basically level them, those cells, and under the microscopy we can even, without killing anything, we can measure how the volume of the, you know, that solid tumor increasing, because we can scan every day. Like, you know, you anesthetize them, keep under the microscope for 10 minutes, scan it, put it back, again do this. So we can really do it. We don't need to take it out anything.
Okay, yeah, I think there's one question from the back there, yeah.
Good afternoon, sir. My name is Subarna Lama. So whenever we are doing any type of testing or evaluation, one of the crucial parameter is temperature. So whenever we are simulating something, so zebrafishes are mostly cold-blooded fish. So my question is, how did you overcome that? Like, are they able to survive in even body temperature that we have?
Very good question. Yep. So we just increased, not keeping at 28 degrees centigrade, we just increase up to 32 degrees centigrade temperature. So we just compromise over there. We can't go beyond 32 degrees centigrade temperature. What happens as the tumor growth, exactly sir mentioned, so then at the 32 degrees centigrade tumor growth will be not as high as 37 degrees centigrade, but you'll get the in between. So yeah, so we can't go, that is, yeah, that we have to compromise. So we just make it 32 degrees centigrade.
So the physiological processes might differ due to the temperature.
So this physiological process, the heart rate will be around 5 to 8% more. The rest of the things fine, they can survive and they are good with that. We don't that model is available, so you can simply take that model and then keep into the well.
Okay.
And for the that Alzheimer, people do the same mouse people does, that treating with the chemical.
Yeah, but in India actually transgenic mice is very rare, people using like that. So is it feasible in your lab, like out of this?
No, if you tell that, okay, you want to work on Alzheimer and you know the target, then one can manipulate the target. So because we do the transgenesis and the knockout regular work.
Okay.
So genetic modification we can do if you know the target.
Okay, thank you, sir.
Hello, sir.
And 2A, this protein loaded this one and then stick it. And then it stay and slowly release of the drug. So you can do this, you can load your drug into that and then. Model has its own advantage and disadvantage. So wherever you find the advantage that we can use with that model and go to the next.
As we wrap up this final technical session, let us take a moment to appreciate the wealth of knowledge we have gained today, from thought-provoking discussions to groundbreaking insights. It has truly been an enriching experience. A big thank you to all our esteemed speakers and participants for making this session both informative and engaging.
The lunch break starts from 12:45 at the same venue near Shivaji Foyer. And please reconvene by 1:30 for culturals and valedictory session. Thank you all.
song.
Sir, on public demand.
Sir,
Will you please?
public demand.
Yes, so my dear students, yes. So today, no, actually, I'm feeling bit, yes, tired. Okay, so. Oh.
Good afternoon, everyone. Welcome back. Now we will continue with the session.
Now we cordially invite our Dean, Professor Jagatran Das sir, to come onto the dais for the closing remarks.
Good afternoon, everyone. I think you had a very good lunch and a nap also, maybe about 10-15 minutes. So anyway, in the closing remarks, I would just like to say that distinguished delegates, esteemed speakers, participants, and colleagues, as we are close to the conclusion of the conference on integrative trends in pharmaceutical and healthcare sciences, Healthcare Next 2025, I want to express my deepest gratitude to each of you for your invaluable contributions. Over the past 2.5 days, we have explored a wide range of scientific topics, engaged in thought-provoking discussions, exchanged innovative ideas, and delved into the cutting-edge trends shaping the future of healthcare and pharmaceuticals. This conference has highlighted the significance of collaboration and integrative strategies in addressing the challenges of our rapidly evolving healthcare landscape. From advancements in drug development to the integration of technology in healthcare delivery, it is clear that the future lies at the convergence of science, technology, and medicine. I'd like to extend my sincere thanks to our distinguished speakers for their insightful presentations and stimulating discussions. Your expertise and vision have been truly inspiring, and your active involvement and enthusiasm have played a vital role in making this event such a successful event. I would also like to extend my appreciation to the organizing committee and all those who work tirelessly behind the scenes to ensure the smooth execution of this conference. Without your dedication and hard work, this event would not have been possible. Additionally, the generous support from our corporate partners made it possible to successfully organize the event. Thank you once again for being a part of this memorable event.
wish you a safe journey back home and hope to meet you once again in some other occasion. Thank you very much.
Thank you, sir. Now we're going to move on to prize distribution session. I would like to invite Professor G. Shivakumar, Pratik sir and Ashish sir on the dais.
Now the prize distribution for the oral presentees. First prize goes to all PPT play cheyatledha.
The first prize for oral presentation goes to Preetham Das from CSIR ISCT. Congratulations. The second prize goes to A. Shailaja from Geetham School of Pharmacy. The third prize goes to B. Chandrika Kumari from Geetham University, Visakhapatnam.
Now for the poster presentation. The first prize goes to Isaac Anand from JSS School of Pharmacy. The second prize goes to Dr. M. Srividya Reddy from Prasad Research Foundation.
Can anyone else collect on behalf of them?
The third prize goes to Harshavardhan Singh from Central University of Punjab. Now I would like to invite all the felicitators, all the evaluators for felicitation onto the dais.
Dr. Naresh Panigrahi, Dr. Vijay Kumar Talluri. Prof.
Professor Jeevataparan.
Dr. Sharana Basappa. Dr. Nandan Kumar. Professor Anil Tumkur M. Dr. Durga Prasad.
నేను చెప్తున్నాను నెక్స్ట్ నువ్వు జీవి రాదో చెప్పేసాక నేను అంటే నెక్స్ట్
Dr. G.V. Radha.
Dr. Pawan Kumar Chintamaneni. Dr. Kranti Kumar Pola. Dr. Ramreddy Godala. Guys, no. Professor Kumaraswamy Gandla. Dr. Nitin Yadav. Dr. Bhaskar Darawat. Dr. Bhanukiran Kancharla. Dr. Babani Shankar. Dr. Pooja Mittal. Dr. Divya Reddy S. Dr. V. Muralidharan. Dr. V. Muralidharan.
We want to thank all the faculty and Dean sir, Principal sir, and Pratik sir and Ashish sir. Thank you.
We would like to request all the evaluators to come onto the dais for a group picture. Along with the winners of both the oral and poster presentation. I would like to request all the committee heads and volunteers to come onto the dais for a group photo. But
Because after cultural.
we request all the faculty to be on the stage for the group photo. Also, the Vizag faculty are requested to join on the dais.
All student volunteers assemble on the dais. All student volunteers.
Thank you all the volunteers. Without you, it can't be possible. Thank you so much, everyone.
Now we would like to invite Ashish sir to deliver vote of thanks.
So, very good afternoon to all, and I'm just saying thanks to everyone, first of all, and as we come to the conclusion of this Healthcare Next 2025, it is both an honor and privilege to extend a heartfelt vote of thanks on behalf of Geetham School of Pharmacy. First and foremost, I would like to express my sincere gratitude to our distinguished chief guest, Dr. Akela, founding director of Dr. Reddy's Life Sciences, Dr. A. Ramkishan, deputy director of the CDSCO, Hyderabad, for gracing the inaugural session with their inspiring words and invaluable insights, particularly how the pharmaceutical and health sciences together improving the healthcare all over the globe. A special thanks to our President, Honorable Vice Chancellor, our Pro Vice Chancellor, for their constant support and encouragement in making this conference a grand success. We are deeply indebted our keynote speakers, expert panelists, including Professor Parimal, Professor Parang, Professor Vinod Kumar, Professor Yura, Professor Amita Verma, Professor S. Jiva, Raghava, Professor Sandeep Bharate, Professor Dr. Uday Saxena, Professor Ganga Raju, and many others, for sharing their cutting-edge research and innovative perspective in healthcare and pharmaceutical sciences. Your contribution have undoubtedly enriched our knowledge and opened new avenues for the future research and collaborations. I extend my sincere thanks, appreciation to the session chairpersons, panel moderators, and judges who played a crucial role in facilitating meaningful discussion and evaluating the oral and poster presentations. A big thank you to all the researchers and participants who presented their work, shared their experiences, and play a crucial role in facilitating this contributed to the academic discourse. Your enthusiasm and commitment to the scientific advancement are truly commendable. To our dedicated organizing committee, faculty members, student volunteer, and technical staffs, this conference would not have been possible without your relentless effort, meticulous planning, and seamless execution. Your hard work behind the scene ensured that everything ran smoothly over the past 3 days. I would like to acknowledge our sponsor and industry partners for their generous support in making this conference possible. Your collaboration together strengthen the bridge between the academia and industry, fostering the innovation and growth in healthcare. Finally, to all the attendees, thank you for your active participation throughout, provoking discussion, unwavering enthusiasm. Your presence has made Healthcare Next 2025 a truly memorable and impactful event. As we bid farewell, let us carry forward the insight and collaboration we have gained here and continued our journey towards the advancement in healthcare and pharmaceutical sciences. Wishing you all the very best luck, and thank you to everyone.
Thank you, sir.
Now for the most awaited part of the day, culturals.
Welcome back to day 3 of culturals.
Let's start day 3 with something timeless.
Shruti's classical performance.